Welcome. Thank you for joining us. I'm Dr. Natalia Wagemans, Global Head of the Purina Institute. Today, I'm pleased to introduce you to the Microbiome Forum virtual event, Putting Microbiome Science into Clinical Practice, organized by the Purina Institute in collaboration with the top experts in microbiome, gastroenterology, veterinary medicine, and nutrition. Before we start our scientific program, I would like to introduce you to the Purina Institute. The Purina Institute is a global professional organization that serves as the voice of science for Purina. Representing a diverse team of more than 500 scientists and pet care experts across a worldwide network of research and developmental facilities, the Purina Institute believes that science is better when it's shared. Through global collaboration, support the pet health initiatives and conferences and events like this webinar today, the Institute shares insights and discoveries about proven nutritional science. Nutrition influences every aspect of pets' life. As a part of the world's largest food and nutrition research organization, the Purina Institute aims to put nutrition at the forefront of pet health discussion. The Institute does not discuss products, only science. The Institute provides veterinary professionals like you with the science-based facts and information you need to have nutritional conversations with pet owners that can help dogs and cats live better, longer life. Now, you know a little bit about the Purina Institute, and if you would like to take more information, you can join us on the purinainstitute.com website and subscribe for our newsletter to get the latest nutritional updates. What is about the microbiome? Nestle and Purina are the leading organizations in the microbiome research. As a part of Nestle, Purina dedicated a long time to research in microbiome, prebiotics, and probiotics to evaluate potential probiotic strengths of value to veterinary medicine and pet health. Nestle has collecting bacterial strains since 1970s. The Nestle culture collection housed in Lausanne, Switzerland, contains thousands of bacterial strains that have been genetically sequenced and cataloged. Nestle published the first bacterium species genome sequence as well as the first lactobacillus species sequence from the human gut. As a pioneers in microbiome research to develop nutritional interventions that benefit pet health, Purina and Nestle made a certain discoveries that helps the veterinarians and pets to be better. For example, first direct comparison of the dog and human microbiomes to demonstrate the similarity between the microbiomes of two species as well as the similarity in their response in diet. Purina offered the first shelf-stable probiotic supplement, SF68, proven to promote a healthy immune system and provide dietary management of dogs or cats with diarrhea. Purina offered the first shelf-stable probiotic to be proven to help with anxious behaviors to dogs better cope with external stressors. And the event today is taking a time to look at the different aspects of the microbiome, science-based evidence, and how to put this into the clinical practice. As you could notice that we have two streams that you can join freely depending on the topic of the lecture. In some of lectures, you will see more academic aspects of the microbiome research, while in the other lectures, you will see more clinical and practical implications. At the end of the session, stay with us because you will have a unique possibility to join a questions and answers session with a panel of the experts who were building this program together and who will share with you science. You will be able to type your question online, and if you know whom you would like to address these questions, please indicate the name of a speaker. That will facilitate our conversations. And now I have an absolutely great pleasure to introduce a chairperson of this microbiome forum who put that program together with the eminent speakers and experts in area of microbiome, Professor Jan Suchodolski. Professor Jan Suchodolski graduated with a veterinary degree from the University in Veterinary Medicine in Vienna, Austria. After working for several years in a small animal specialty clinic, he returned to academia and received his PhD in veterinary microbiology from Texas A&M University for his work on molecular markers for the assessment of the intestinal microbiome. He is board certified in immunology by the American College of Veterinary Microbiologists. He currently serves as professor and associate director in the GI lab in Texas A&M University. He has published more than 340 peer-reviewed manuscripts in the field of veterinary gastroenterology and microbiome. And with this, I welcome Jan to give his keynote lecture on part of the whole, the role of the gut microbiome in the host health. Jan, the floor is yours.
Yeah, welcome everybody. It's my really great pleasure to be here. And so my talk will be really as a starting point and really to give an overview of all the different talks that's going to come. And it's about the part of the whole and why the microbiome really plays such an important role in host health, but also disease. First, my disclosure slides. I'm an employee of Texas A&M University, and we actually offering microbiome testing as a fee for service. But I'm also consulting or speaking for different companies. So why is the microbiome important? I think more and more over the last years, we discover that really gut bacteria play an important role. And some of them are really surprising. Like, for example, here's an example of a recent publication in a very high impact journal. And they've shown that some people that have so-called fatty liver disease. So it's a fatty liver disease. It's one of the most common disease phenotypes, liver diseases in human medicine. And it has been shown that some of those people have actually bacteria that can produce ethanol. So if they're on a high carbohydrate diets, they have some strains, they take those carbohydrates and ultimately convert them to ethanol. And that is believed to some degree contribute to this fatty liver disease. And even in the study, they really showed even nicely that if you take feces or fecal samples from some of those patients, put them into a mouse model, and really you could induce liver disease in those mice. So again, showing that the gut microbiota plays an important role in some disease phenotypes that we until now didn't really well understand. On the other side, also like treatments that we usually always thought that they are to some degree beneficial or at least harmless. So getting me wrong, antibiotics are super important for certain diseases. But there's more and more data coming out that antibiotic treatments, especially with broad spectrum antibiotic, can contribute to some chronic diseases that were now very prevalent. So for example, in this really huge study from Sweden, looking at several 120,000 patients, they showed that exposure to antibiotics is potentially associated with an increased risk for developing GI disease. So again, we have to overthink a little bit the benefits and the risks of antibiotic administration. There seems to be also some data out there that shows, especially in young childhood, in a certain young age, when the microbiome develops, when the immune system develops, antibiotic exposure also is a risk factor. It's a small risk factor. So again, I'm highlighting there's no right or wrong answer, but it just shows that when we use antibiotics, we most likely change the gut microbiota. And then again, it becomes a part of a disease phenotype. So again, we're learning more and more that the microbiome is clearly implicated in beneficial compounds, but also potentially, if changed, can be a contributor to disease. And why is that actually? Because really surprisingly, what we always thought, this is a waste product. So when you look at the GI tract and we look at a fecal sample, we always thought that's a waste product. But actually, it turns out that this is an important metabolic organ. It's immune, but also metabolically active. And the reason for that is if you cut through the lumen of the colon here, for example, that's in a mouse, but the same we see in humans and dogs and cats. What we see here in blue, that's epithelial cells, so that's the host. And here in red, all those cells here in red, those are bacteria. So macroscopically, what we see as a fecal sample is actually really full of bacteria on other microbes. and they are really alive and they're metabolically active. And so we need to understand that the gut microbiota is really part of our physiology. So everything, when you think about it, everything what's going to enter the gut. This could be nutrients, in macronutrients, minor nutrients, but also drugs, medication. Everything what enters the gut is to some degree going to be changed or altered or metabolized by bacteria. And again, along the entire GI tract, those metabolites can be ultimately absorbed by the host. And depending on the balance, so the balance of the microbes and the balance of what they're actually producing, we really have either pro-inflammatory or anti-inflammatory signaling. Ideally, it's obviously both. So again, it just shows us that this is important. But also what we're going to learn today a lot, and I think we really want to really emphasize that, we cannot separate the microbiome from our organisms. So very often what happens in the host, there could be inflammation, there could be other factors that's going to change the gut microbiota too. And again, so what we really need to understand is we cannot treat the microbiome by itself. There's always a reason gut microbes are changed. It could be dietary, but could also be some inflammatory components. And so ultimately, this is all one big organ. And for the future, multimodal approaches are really crucial. And that's why I think this connection, there's more and more data coming out that the guts and the gut microbiota really play a big role, obviously for health, but if there are alterations, that could be, again, different dietary substrates, but also some underlying disease process in the host that changes the gut microbiota. And ultimately, we have this really important axis between the gut microbiota, the circulation. We can have it, for example, in the kidney and so on. So again, we need to understand everything is going through the gut to some degree, and this becomes really a central role in many disease phenotypes, not just for the intestinal tract. So what I'm going to briefly talk today is really, what is the microbiome? Mostly focused on a few metabolites, because I think the more you read in literature, it seems very complicated. Yeah, every publication finds something. But I think it's like any other organ systems. If we reduce it to a few major metabolic pathways, and those are really the major ones, like the short-chain fatty acid pathway, amino acid metabolites like indoles and so on, and especially bile acids, I think that probably explains a lot of those effects of the microbiota on the host. I'm going to touch up also a little bit of dysbiosis and actually really what is the most common reason, and that's really GI disease, underlying GI inflammation, or obviously antibiotic usage. So to understand that those are really the major factors, and trying to really highlight a little bit the relationship with the microbiome, dysbiosis, and systemic organ disease. So again, I think when you read literature, a lot of people say, well, it's so complicated, and it truly is. I'm sure it's like every other organ system. But I think when you really start looking at this in detail, there's a lot of pathways that really repeat itself. And again, I mentioned the short-chain fatty acid pathway is one important pathway. That's actually a pathway we knew for a long time. We have carbohydrates from the diet, mostly complex carbohydrates. And really, some key bacteria, and again, it's also important to probably start focusing on a few key bacterial species, because again, once you start reading literature, they're going to keep coming up. So it's really not as complicated as we may think, as long as we focus on one of those major pathways and major key bacterial species who do some of those functions. But important is that some bacteria really convert carbohydrates from the diets into so-called short-chain fatty acids. And again, also important to highlight, many of those metabolites really also depend on how much it is that. There needs to be a balance. In proper amounts, they're very highly beneficial. They're anti-inflammatory, and they also maintain intestinal barrier function. However, in abnormal concentration, if the ratios are different, then also they can actually activate some entropathogens. One important pathway is the tryptophan pathway from the diet. It converts by bacteria into so-called indole metabolites. And generally, again, it's anti-inflammatory, important for barrier function. But again, if it's in abnormal concentration, it can actually act as an uremic toxin. And the bile acid pathway, a very crucial one. And this is also one key species that we're discovering now in dogs and also in cats. It's really beneficial, it's Clostridium anonis, because it transforms those primary bile acids in the GI tract into so-called secondary bile acids. And again, in beneficial concentration, they're anti-inflammatory, inhibit pathogen growth, and so on. But again, if they're abnormal, they can either cause diarrhea or in too high concentrations that can also damage the mucosa. So again, everything is a balance. So a few examples, how do those metabolic pathways really work? So the short-chain fatty acid pathway is well established. I think we knew it for the longest time. And some of the short-chain fatty acids really, they bind to so-called G-coupled receptors. And those ultimately are anti-inflammatory. They can signal and can really improve barrier function, amongst others, so many different pathways. And again, Faecalibacterium, for example, is a very important beneficial species in dogs and in cats. There's also new features that we discover. Yes, so we know that those short-chain fatty acids bind to some of those receptors, and ultimately, if they activate many immune modulatory compounds, so generally, it is a beneficial pathway. But we're also discovering that that's why the microbiome becomes so central in systemic health, is that some of those receptors, for example, are now also discovered to be on other organ systems. So we always think about the gut, but as you can see, many of those receptors are also on the kidney and so on. Another important pathway, tryptophans. Again, as I mentioned before, tryptophan comes from the diet, gets converted by bacteria into different indoles, and generally it's a very important pathway. I think in GI disease, we're discovering more and more, if there's decrease in tryptophan, there might be some association that supplementing tryptophan, at least in mouse models, may be useful. However, again, balance is important. In certain conditions, bacteria can actually convert those indole pathways in so-called toxic metabolites like indoxyl sulfate and so on, with systemic negative effects on the host. So again, it depends always on where the host is. And again, the bile acid pathway is really one of them that is really surprisingly one of the probably key pathways. And the reason for that is that primary bile acids come from the liver, usually aid in fat digestion. Most of those bile acids get reabsorbed back in the liver for enterohepatic circulation, but a small percentage of bile acids gets actually converted by bacteria, so gut bacteria, into so-called secondary bile acids. And this is a beautiful example to show that really how microbiota is part of our physiology, because to our current knowledge, only bacteria can perform this conversion. So only bacteria, in this case, it's Clostridium anones in dogs, also in cats, convert primary bile acids into secondary. And as you can see, there's a lot of effects. The proper ratio of those bile acids is crucial for signaling back to the liver. So for liver homeostasis, there's a lot of receptors like the TGI-5 receptor, for example, that is located in many different tissues. If it gets activated, and mostly those secondary bile acids are the strong activators, activation really downregulates pro-inflammatory signaling. We also know that's also important. Many of those metabolites really regulate the pathogens. So often we really see pathogens increased in GI disease, and we think that's the cause. But actually, importantly, because we have a dysbiosis, we have some of these metabolic pathways disrupted that ultimately leads to an overgrowth of pathogens. And finally, we also have, especially bile acids are very important also for insulin glucose secretion and so on. So again, as you can see, a pathway that is regulated by microbiota has many different systemic effects across the whole body. Here another example why bile acid receptors are important. As you can see, they activate different receptors and play a major role in glucose homeostasis, lipid metabolism, in motility, gallbladder filling, and so on and so on. Again, it's really a very important microbial pathway that has a lot of systemic health implications. And this is actually one we looked for several years now in the bile acid pathway. And this is work from Paula Garretta. She was back then a PhD student and a pathology resident. Now she's a really successful pathologist in Brazil. And what she did, she actually in dogs, in healthy dogs, along the GI tract, stains for this important TGF-5 receptor. And you can see this is highly expressed in many different tissues, in goblet cells, colonocytes, macrophages, endoendocrine cells, ganglion cells, and so on. So again, clearly indicating that those receptors that are activated by bile acids are highly abundant in many different organ systems. So this is really a key pathway. And that really brings us also that, of course, it makes sense that we'll need to start looking at the microbiome. We need to understand, is it normal, abnormal? And obviously, when you look at the GI tract, it's a very complicated, long organ. So that's why it's also very difficult to truly appreciate the full microbiome along from the small to the large intestine. So there's really not one single test that allows the proper assessment of microbiome function. However, with time, we really, I think, we're able to, with many different studies and many different studies over time, we were really able to understand that some key pathways and also some key bacteria who do those pathways. So I mentioned, for example, Fecalibacterium, Clostridium rhamnosus. So we developed assays for those. So we can really measure the abundances, and we actually can even describe reference intervals for those. And so we have developed a test called the dysbiosis index, who is really measures 7 bacterial groups. 5 of them are called beneficial, so they usually go down in disease. And some of them are more or less harmful, not necessarily directly harmful, but they are usually increased in dysbiosis and so on. And so we can really measure them. We can report their reference intervals across a huge population of healthy dogs around the world. We have a lot of different studies about that. But also more importantly, we can really combine the results into one single number. And so what you see here is the dysbiosis index. What you see here, each dot here is really the combined results of all those 7 abundances. And what it can tell us that we can really define to some degree where most healthy dogs lie, and they're below 0. Between 0 and 2 is probably like a small shift, a few healthy dogs later. And then above 2 is actually we're quite specific for that's an abnormal shift in the gut microbiota. But also we're discovering now that we need to kind of, interpretation is that we look at individual groups. If some of them are abnormal outside reference interval, that is a minor form of dysbiosis, and the index itself indicates how all those groups shift together. And we're also discovering more, and Dr. Torresen is going to tell you a little bit more about it. The higher the dysbiosis index, the higher the microbiome shift, usually it's a very negative predictor for treatment outcome. And because again, there is a lot of changes in the gut environment, and the microbiome really, dysbiosis reflects that in an increased dysbiosis index. So what are really major changes? So again, I mentioned antibiotic usage. The more we learn about studies, we need that antibiotics are really sometimes very useful, obviously, in many cases situations, but they definitely have an impact on the gut microbiota. So with this dysbiosis index, because it's a very highly reproducible test, we can really compare different studies, and we can really follow up patients over time. And you can see that different antibiotics like tylosin, metronidazole, broad spectrum ones, truly increase a huge shift in our gut microbiota. Most animals are going to come back over time within 1 or 2 months, but there's always a few animals that are going to stay abnormal. So in some animals with antibiotic usage, we can potentially induce a long-lasting change in the gut microbiota, especially in some of those important groups like Fecalibacterium or Clostridium rhamnosus. Also in cats, as you can see, there's an assay we just finished validating. The paper is submitted. Similarly, below 0 is healthy cats. A lot of cats with chronic enteropathy have increase in the dysbiosis index, again, decrease in Fecalibacterium and so on. And again, also in cats, antibiotic is a huge impact on the gut microbiota. So again, those are 2 major fronts. Chronic GI disease and antibiotics have a huge impact on the gut microbiota. And why is actually GI disease so important? Well, think about it. This cartoon kind of illustrates a little bit how the microbiome is organized. So in a healthy gut, we should have a very well-organized microbiome, highly diverse, many different species. And they are separated from the epithelial cells by this mucus layer here in yellow. So this mucus layer separates the gut microbiota from the epithelial cells. Then we have a well-organized epithelium, and that's why also a normal functional immune system. Now, often when we look at GI disease and biopsies, we kind of always very much focus a lot on the infiltration. Is it what kind of cell infiltration is? But if you look really closely, we also need to understand that in GI disease, inflammation is important, but also the architecture of the GI tract is very, very different. It means we have shortening of villi, we have loss of brush border, and so on. So again, it's, I think, important to understand that once a chronic GI disease has developed, it's not just the inflammatory component that's important. There's a lot of changes. And many of those changes we also discover now take a long time to normalize, if they normalize at all in some patients. So as you can see, in GI disease, we have always a dysbiosis, not always, in many cases we can see a dysbiosis. In some patients, but not all, we have a decrease in this mucus layer. We have that because of that, we have attachment in some patients of bacteria to the epithelium. We have rupture of the epithelial tight junctions and so on, and an overshooting immune system. So again, in chronic GI disease, it's just that one part. There's a lot of different things that can go wrong, Many of them are very long lasting, so the function of the GI tract is abnormal. And that's what is really also drives dysbiosis. So it's not just one part. We need to always see this all together and ideally treat this all together. And so here's an example of fluorescence in situ hybridization in a healthy dog. Here you can see in red, that's the bacteria. And you can see in this black layer here, that's the mucus. This mucus is very, very important because it really separates out or the bacteria from the host epithelium. So that is a normal gut. In some dogs, but not in all dogs with chronic GI disease, it can look like this. Here in blue, you see epithelium. Then you don't see this mucus layer anymore. So we have a lot of bacteria that are attached to the epithelium, obviously inducing inflammatory components or pro-inflammatory signaling. So again, the problem is also a little bit that not every dog looks like them. So along the entire GI tract, a lot of different features, but understanding that many compounds in chronic enteropathy are wrong can help us ultimately thinking about what diets are needed. And again, important is that really, this is, I think, one that we probably, this functional aspect, once an abnormal GI tract is important to understand too, because once we have malabsorption, so this is again, the villus of a healthy dog. This is the work from Paula. And here in brown, you see the bile acid transporter in the ileum, and this one is responsible for absorbing of bile acids. And you can see how superficial these transporters are. And that's sort of the brushboard. And it's not just about bile acids. Here's absorption of, for example, glucose, fatty acids, and so on. So that's like really in a healthy dog. In a diseased dog, so many of this brush border is gone. That means we have also a lot of malabsorption to some degree. Many of those compounds ultimately are going to be malabsorbed. They stay in the lumen of the GI tract, and that really also drives the dysbiosis pattern because bacteria like to feed on them. So again, understanding it's all a combination. There's not just dysbiosis. Dysbiosis in many cases really the reflection what happens in the gut environments. The microbiome is probably the first one who really reacts to changes on the brush border, inflammation, and so on, and often can be one of the first markers that there's something abnormal in the GI tract. And then ultimately, if this is abnormal, the gut bacteria can contribute to diseases because, for example, if there's glucose malabsorption in some patients, so this is in fecal samples, we see that some patients with chronic enteropathy have increased glucose, for example, bacteria obviously can metabolize it and can cause to some degree substances that again cause diarrhea. So again, there's a host part and microbiome part. We need to see them both together and ideally treat them together. And again, this is really probably my last slides for the moment for the first talk to understand, because I think we often think about the microbiome by itself. We think we can treat the microbiome by itself. Often we think about pathogens, we think that is the cause, but really what happens often, this interface between the host, the epithelium, and bacteria, that's where a lot of problems are starting. So often what happens in the host, inflammation can cause dysbiosis. Dysbiosis again can contribute to clinical signs of further GI disease. Then we have all of those components, changes in pathogens and so on. So understanding is that we need to think about when we see dysbiosis or enteropathogens, understanding that in most cases, the dysbiosis is a central part, but often, again, it's probably a consequence of those underlying disease processes here. And treating the underlying disease process is crucial. So it's difficult to treat just a microbiome. We need to treat the underlying condition, and usually not everything's going to normalize. So again, I think to summarize, and really many of those principles going to be discussed by some of our speakers later on, is that the microbiome is obviously very important. We need to see the microbiome as a metabolic organ, and that's why there's a lot of systemic health effects. So obviously it's important to keep the microbiome healthy. At the moment, we probably learned more than prevention is much better than therapy. But generally, we also need to understand we cannot just treat the microbiome by itself. We need to understand that dysbiosis is often the first mark of an underlying disease process that we need to identify and treat that, because if we just treat the dysbiosis, it's going to keep coming back because, again, it's the reflection of abnormal gut environments. But ultimately, we're going to discuss today from many speakers, what are pros and cons of diet, why diet is probably one of the most important reasons should always be considered dietary changes when we see an altered microbiome or dysbiosis. What are the benefits of prebiotics and probiotics? We're going to discuss Dr. Unter is going to discuss the judicious use of antibiotics. Antibiotics are clearly needed in many diseases, but we need to understand in many, they're not important and might potentially have a risk factor. So weighing the benefits versus risks. And also we're going to discuss that actually fecal transplantation, we know a little bit more about in the last few years. There are a few case series, case reports, some retrospective studies, but there's also a lot of prospective studies ongoing. And I think we're going to see that fecal transplantation is going to be an option in some of those patients. Not in all, but it's definitely one option that we can consider in the future. So with this, I want to just acknowledge many of my collaborators that are really helpful in collecting all this data and understanding how the microbiome works. Without it, it would not be possible. Obviously, grant supports, and I'm happy to answer any questions.
Our next speaker is Dr. Linda Torresson. Dr. Linda Torresson graduated from the Swedish University of Agricultural Science. She has worked at Evidensia Specialist Animal Hospital, Helsingborg, Sweden, focusing on small animal gastroenterology. In 2001, she became a Swedish specialist in disease of dogs and cats, and in 2007, a Swedish specialist in small animal internal medicine. For a few years, she served as the medical director at Evidensia Specialist Animal Hospital, Helsingborg, where she is combining clinical practice with GI research. Her PhD was dedicated to cobalamin supplementation in dogs with chronic enteropathies. Her main research interest is the intestinal microbiome, including modulation of the microbiome beyond antibiotics. Dr. Torresson currently serves as the president of the European Society of Comparative Gastroenterology. Please, Dr. Torresson.
I would like to thank the organizing committee for inviting me. These are my disclosure coming up here. So this is going to be a very clinically based practical lecture, all centered around cases I've seen where the microbiome is involved. So let's start with meeting Jycke. So Jycke means doggy in English, and he's a wonderful mixed breed dog, and he was 7 years of age the first time I saw him, an intact male. And for over a year, he's had attacks of abdominal pain, lethargy, and recurrent diarrhea. So they only took him to a vet. The vet took fecal samples, found hemolytic E. coli, and treated him with amoxicillin according to culture and sensitivity testing twice. And that didn't really help. So they only took him to another vet at another clinic. And they took new fecal samples. The samples were negative for parasites. And guess what? He was still positive for E. coli of the hemolytic type. So now he had the third course of amoxicillin, and it didn't really help either. So now they went for endoscopy, and the biopsies showed a mild diffuse lymphocytic plasmacytic gastritis with presence of mild to moderate amount of Helicobacter-like species, and also a moderate to severe lymphocytic plasmacytic chronic enteritis. So now he was treated with amoxicillin, metronidazole, and a proton pump inhibitor. And that didn't really help him either. So I'm sure this was done because they wanted to treat the Helicobacter-like species, but there is very little, if any, evidence to use this triple combination to treat that. And really, we don't expect a dog to get that degree of diarrhea with only Helicobacter-like species, because that's a very, very common finding in gastric biopsies and something you most often can overlook. So now he was sent for referral, which is when I saw him. This is a picture from 2 weeks ago. He's now 13.5 years old, and he's an absolutely marvelous dog. So when he was 7, I started treating him with a hydrolyzed soy-based protein diet. He was on an intestinal diet when I saw him the first time. I started a probiotic and a prebiotic, and we're going to discuss that a bit later. I also started immunosuppression with prednisolone, and he responded really well to that. But we couldn't taper the dose to acceptable maintenance doses because he had new flare-ups. So we added a second-line immunosuppressive. And first I tried azathioprine, which really didn't help at all. And cyclosporin helped slightly, but he still had those flare-ups. But mycophenolate has been really beneficial for him. So now he has flare-ups, but really rarely, like once every 6 months, and sometimes not even one for a year. And they are very, very mild compared to what they used to be. So he's not being grumpy any longer, he's having a good quality of life, still very playful, but he's had 4 courses of unnecessary antibiotics, because the antibiotics he was given, they really didn't help him at all. So if we're going to investigate dogs with acute or chronic diarrhea, should we exclude a bacterial infection of the GI tract with a regular culture? So we can take Q-tip and do a rectal swab, or take the whole fecal sample and submit for culture and sensitivity testing. And we can do that, but we will get very limited information. Because the intestinal microbiome is a very complex ecosystem, and the biggest problem regarding culture and sensitivity testing is that only about 5% is detected with a conventional culture. So with modern molecular tools, such as the 16S rRNA sequencing, up to 450 different bacterial species have been found just in the jejunum that used to be thought of as a more sterile place. So if we take a culture, we can find out virtually what's happening in about 5% of the whole microbiome, but the remaining 95%, we don't have a clue what's going on. And one of the biggest reasons for that is that the microbiome mostly consists of anaerobes, and they are really difficult to culture, such as Faecalibacterium. Faecalibacterium is very important for gastrointestinal health. It can make up to 16% of the total microbiome, but it's very difficult to culture. And Faecalibacterium produce a lot of metabolites that have a beneficial effect of the host, such as short-chain fatty acids that can have an anti-inflammatory effect. So the old belief that if we have gastroenteritis, we can find one single pathogen and kill it with antibiotics and everything will be fine. This is very outdated. The biggest difference in acute and chronic gastroenteritis compared to in health is that the good ones go home. So we have a less diverse microbiome, and we have a significant reduction of the beneficial microbes, the good ones. So we have decreased production of short-chain fatty acids, and those bacteria typically are Blautia, Faecalibacterium, Thericibacter, and Fusobacterium. And this was first shown in a paper from 2012 by Ansa Godolsky and co-workers. So we can actually think about a party instead, if we're going to think about what's happening in the gut. So we can have a good party, or we can have a bad party. This is the good party. This is a Midsummer Eve party, actually from this year, with me and my friends. So at the good party, there's diversity. There are lots of different bacterial taxa, and there's also food. Food in the gut is short-chain fatty acids that are produced by the beneficial microbes, and that can act as an energy source for the colonocytes. At a good party, we have a nice atmosphere and also an anti-inflammatory atmosphere if we think about the gut. And that's produced by short-chain fatty acids, indole and secondary bile acids that have an anti-inflammatory effect in the environment of the gut. We also have a buffer, so if there's someone nasty coming to this party and I don't want to talk to this person, there are lots of other people I can talk to. And that's the same thing in the gut. If you have a buffer and there's one nasty one coming down, then it really doesn't matter that much because there are other ones that will take care of it. At a good party, there's no leakage. So if we discuss something sensitive, it's going to stay around the table. And in the gut, the no leakage is produced by short-chain fatty acids and indole because they enhance the barrier function and they also improve the tight junctions. A good party is also characterized with healthy motility. So after this meal, we'll go for a long walk or start dancing or something like that. And in the gut, the healthy motility is helped with short-chain fatty acids as well. A good party in the gut also have control of proliferation of negative bacteria, and that is helped with secondary bile acids. About 95% of all bile acids are reabsorbed in the ileum, and about 5% will enter the colon. And there they will be converted to secondary bile acids with help of Clostridium hierinonis. And the secondary bile acids have an anti-proliferative effect on E. coli, Streptococcus, Clostridium perfringens, and Clostridium difficile. So that's the efficient cleaning up effect at the party, that Clostridium hieronymus can convert primary bile acids to secondary bile acids. So now we have the bad party. We have less diversity. The good ones have gone home, so we have less buffer capacity if something nasty happens. We have a lack of food. There's no one producing short-chain fatty acids any longer. There's a toxic atmosphere, so we have a pro-inflammatory atmosphere because we are lacking the metabolites produced by the beneficial microbes, and we are also lacking secondary bile acids. We have increased leakage, so the tight junctions won't work as well because we are lacking indole and short-chain fatty acids. And we have an accelerated motility because we have a lackage of short-chain fatty acids and we have increased amount of primary bile acids. They will draw water into the colon and also accelerate the motility of the colon in a negative way. And we will also have increased proliferation of E. coli, Clostridium perfringens, and Clostridium difficile because we are lacking secondary bile acids. So this theory that we shouldn't culture dogs with chronic enteropathy, has this really been proven? There's a very nice study from last year by Melanie Werner and co-workers, where they took fecal samples from 36 dogs, so 18 healthy dogs and 18 dogs with chronic diarrhea. And they split those samples and they sent them to 3 different labs. The labs didn't know if they looked at healthy samples or samples from dog with diarrhea. And they also submitted samples for dysbiosis index. And the fecal cultures failed to distinguish between chronic diarrhea and healthy dogs. Clostridium species were as prevalent in healthy dogs as in dogs with chronic diarrhea, and there was a high level of inter-laboratory variation for culture. If we look a bit more in detail on this is a graph from this paper, and you can see lab 1, lab 2, and lab 3, and the green arrows point to lab 1 and lab 2. So healthy dogs are the red bars and chronic diarrhea dogs are the black bars. And the pale part of the bar represent what is reported by the lab as an abnormal microbiome. So if we look at lab 1 and lab 2, so follow the arrows, then you can see that there are actually more healthy dogs being reported as having an abnormal microbiome by the lab, and this is based on culture. So the culture really can't tell us if it's from a healthy dog or a dog with chronic diarrhea. Growth of hemolytic E. coli could not differ chronic diarrhea from healthy dogs either, so that didn't really help. But the dysbiosis index could significantly differ healthy dogs from dogs with chronic diarrhea. So we see dogs with chronic diarrhea to the left and the healthy group to the right, and there's a significant difference. The dysbiosis index should be below 0 in healthy animals, and as you can see, many of the dogs with chronic diarrhea have an increased dysbiosis index. The green dots represent depletion of Clostridium hieronymus, the bacteria that converts primary bile acids to secondary bile acids. Furthermore, there is a significant difference in presence of Faecalibacterium and Fusobacterium in dogs with chronic diarrhea and healthy dogs. So healthy dogs have higher amount of both Faecalibacterium and Fusobacterium compared to dogs with chronic diarrhea. The dysbiosis index is made up of several different parameters. So the total amount of bacteria, Faecalibacterium, Thericibacter, E. coli, Streptococcus, Lautzia, Fusobacterium, and Clostridium hieronymus. And this was first presented in a paper from 2017 by the GI lab from Texas A&M University. So typically in chronic enteropathy, we have lower amount of total bacteria, lower Faecalibacterium, lower Thericibacter, higher E. coli and Streptococcus, and lower Lautzia, Fusobacterium, and Clostridium hieronymus. So Faecalibacterium, Thericibacter, Lautzia, and Fusobacterium are all short-chain fatty acid producing bacteria, and Clostridium hieronymus converts primary bile acids to secondary bile acids. So can we use this index in clinical practice then? This is Samson, a Basset Artesien Normand, an intact male, 4 years of age when I saw him. So he came as a referral. He has diagnosed chronic enteropathy and only partial response to corticosteroids and a hydrolyzed protein diet. He has borborhythmia, flatulence, grass eating, which is absolutely manic after hunting, and really can't do his job any longer, so he doesn't function as a hunting dog because he can't focus on his task. He has a mild weight loss, and the owner has to stop hunting with him because Samson isn't energetic enough to do that. He did have diarrhea and vomiting prior to starting corticosteroids, and that improved, but he still have those signs of chronic enteropathy. The current treatment when I saw him was prednisolone 0.5 mg per kg every other day and sucralfate 500 mg once daily. On physical examination, he was bright, alert and responsive. He had a body condition score of 4 out of 9 and was very painful and tense in his abdomen when I tried to palpate him. And he vocalized loudly. Routine blood work was within normal limits. His fecal sample showed a dysbiosis index of 2, so dysbiosis. He also has whipworm and borderline cryptosporidium. The ultrasound scan showed that his stomach had an increased thickness of the mucosa layer, and the duodenum and the duodenum also had a pronounced mucosa layer with increased echogenicity and a mild amount of speckles and prominent mesenterial lymph nodes. So he looks like any other dog with chronic enteropathy, basically. This is his dysbiosis index. So he has an index of 2, and he's really depleted of Clostridium hierinonis, 0.1, so very, very low in Clostridium hierinonis. He's also low in Blautia and Fecalibacterium. So he's clearly lacking beneficial microbes. So how can we improve his microbiota? So how can we improve the microbiome in dogs with chronic enteropathy? Let's start with diet. There are 2 different studies showing that diet can induce remission in chronic enteropathy, even looking at the microbiome. So in this first paper from 2019, 29 dogs with chronic enteropathy were fed a hydrolyzed soy-based protein diet. And 20 out of those 29 dogs improved clinically within 14 days. And after 42 days, a decreased abundance of E. coli and Clostridium perfringens was proven in a sample from the microbiota. And they also had an increased amount of Clostridium hierinonis and reduced severity of dysbiosis and increased secondary bile acids. The other paper was published in 2019, and 10 dogs with food-responsive enteropathy and 14 healthy dogs were included. So they were given this animal protein-free diet for 60 days, and the microbiota recovered in the dogs with food-responsive enteropathy. So after having been treated with this diet for 2 months, they had a microbiome that was much more like a healthy dog compared to baseline. Another way to improve the microbiome is to reduce the inflammation. This paper was published this year by Rachel Pilla and co-workers, and they had 9 dogs with steroid-responsive enteropathy that went into remission after 8 weeks of steroids. And they stayed in remission for a year. And they had samples from 5 of those dogs available for long-term follow-up, and they compared that to samples from 13 healthy dogs. So here we see the microbiota of the healthy dogs to the left. That's the nicely colored bar, and the dogs with steroid-responsive enteropathy. So the yellow bar on the right from the healthy dogs represent the microbiota at baseline in dogs with steroid-responsive enteropathy. So you can see that there are huge differences in color here because the microbiome is very depleted in those steroid-responsive dogs. And then we see the same bar after 3 weeks and after 8 weeks. And it's still very yellow. It still really doesn't look normal because the microbiome hasn't really recovered at this point, even if the dogs after 8 weeks were clinically normal. So a significant recovery of the fecal microbiome and the metabolome was seen after a year, and that's the bar to the right. And you can see that it very much looks like the bar to the far left. So the microbiome recovered, but it took much longer for the microbiome to recover compared to when the clinical signs disappeared. So Samson has already been treated with diet and is also been treated with corticosteroids. Probiotics then, is there any evidence to use probiotics to stop or improve the microbiome? This paper from 2013 from Giacomo Rossi and co-workers showed that a probiotic could actually improve gastrointestinal health in dogs with chronic enteropathy. So they had 20 dogs with that was given a multi-strain probiotic, which is at the time was called VSL3 and now is called Microbiome in the US. Or they were given prednisolone and metronidazole. And they could see a significant improvement in both groups. So the canine IBD activity index decreased in both groups. The histology scores decreased in both groups. They took samples, biopsy samples at baseline and after 90 days in both groups. And they could also show that CD3 positive T cells decreased in the mucosa of both groups. But only the dogs that were given the probiotic increased in Faecalibacterium after treatment. So there is evidence that one type of probiotic can improve the gastrointestinal health and the microbiome in dogs with chronic enteropathy. Fecal microbiota transplantation, we are going to discuss that in a later session, so I'll leave that for now. Prebiotics then. The problem with prebiotic is that most studies are done in healthy animals. There's one study from 2019 where a fiber bundle was added to 2 different types of diet, so one hydrolyzed meat-based diet or a grain-rich diet. And those diets were given to healthy dogs or dogs with chronic enteropathy. And after 4 weeks, the microbiome had improved in dogs with chronic enteropathy, regardless if they had the meat-based diet or the grain-rich diet, but both diets had this fiber bundle added. So dogs with chronic enteropathy had improved stool quality, increased species enrichment, and also less loss of amino acids in the stool, a lower pH, and more short-chain fatty acids in the stool when they were treated with this diet with added fiber bundles. And the fecal microbiota transplantation, we'll leave that for later. So back to Samson, what did we do? We dewormed him with fenbendazole. I started him on cyclosporin 5 mg per kg once daily, and I could taper the corticosteroids to lower doses. And I saw him again after 8 weeks, and he was doing really well. He was less flatulent, he had less borborygmia, he was much more alert, and he had increased his body weight with 1.2 kilos. And this is his new dysbiosis index. So the beneficial microbes have recovered. His Clostridium hierinonis is improved. That was severely depleted before. Blautia and Fusobacterium is also improved. He has a slightly decreased amount of Streptococcus, but that really doesn't matter. So he now has normal biosis of beneficial bacteria. Could we now taper the cyclosporin to every other day, possibly? Well, accidentally, he was without cyclosporin for 3 days because they couldn't get hold of it. And that caused severe diarrhea and pancreatitis, and we had to hospitalize him for 24 hours. So basically, he needed the cyclosporin to keep the inflammation in control and also keep the microbiome in control. Next case is Fleesan, border terrier intact bitch, 1.5 years of age with food responsive enteropathy. She has already been diagnosed with the chronic lymphocytic, plasmacytic gastritis and enteritis, and she usually vomits during flare-ups. But now she has acute, profound diarrhea. She has traces of fresh blood. She doesn't vomit, she's still alert, and she has an appetite. The owner is on holiday, so they visited the nearest local vet. And the local vet took fecal samples that were sent to an external lab. And the fecal sample showed that she was positive for Campylobacter jejuni. So the local vet prescribed erythromycin against the Campylobacter and a kaolin pectin probiotic paste. Now, the dog owner didn't really want to use antibiotics, so she didn't give that. She gave the paste. And she tried to contact me, and we spoke after 5 days. So Fleesan still had very soft stool 5 days later, but she didn't have any melena, she was alert, she had a good appetite. The problem now is that the dog owner is going abroad in 4 days' time. So Flisan will be left at a boarding kennel. And boarding kennels really don't like to board dogs with diarrhea. So should we give Flisan antibiotics or not? Well, we tried a prebiotic first. So we gave her psyllium, 5 mils per 12 hours, so twice daily. And the day after she had a normal stool, and we didn't give her any antibiotics. It has been shown in studies from Sweden, and the similar studies are available from Denmark, that 39 to 76 dogs of healthy young dogs below the age of 2 shed Campylobacter jejuni or Uppsaliensis in the feces without having any diarrhea at all. So these are the papers. So you can find Campylobacter in very many young dogs without them having any diarrhea. And Flisan was a year and a half at this time. So possibly the presence of Campylobacter may be due just to her age. They may have been there all the time, and we didn't give her any antibiotics. Last case, this is Hilda, an intact border collie bitch, 3 years of age. She is really healthy. She's a very active dog. She's owned by one of my veterinary colleagues. And she competes in agility, and she does a long distance running with the dog owner several times a week. I've used her as a healthy control dog for 2 different studies, so I know she's a very healthy and active dog, and she has a normal stool and normal stool frequency. And she has a body condition score of 4 out of 9, unless when they have been running too much, and then she becomes a bit thinner. So I want to screen her as an FMT donor because she's a healthy dog, and she is also coming with the owner to the hospital where I work several times a week, so she's an excellent donor. So we're going to screen her. And she has a dysbiosis index of -4.5, and she has a high amount of beneficial microbes. So typically lots of Faecalibacterium and lots of Fusobacterium. So she really looks like a good donor here, but she's PCR positive for Campylobacter jejuni. And this is not dysbiosis. She has a high amount of beneficial microbes, and she is healthy. But she has Campylobacter jejuni as part of her intestinal microbiome. So maybe she may not be the best FMT donor we could think of. If you infect puppies with Campylobacter jejuni, 50% of them will develop mild self-limiting disease. So she may not be the best FMT donor, but she's a healthy dog anyhow. So my take-home message is don't over-interpret results of fecal cultures. Actually, I don't think there's much room for regular fecal cultures in dogs with chronic enteropathies. However, you can use the dysbiosis index to assess the microbiome in chronic enteropathy and also assess the improvement. And also, inflammation drives the dysbiosis. Try to be kind to the microbiome, avoid unnecessary antibiotics, and also think about the One Health perspective. So we really should save antibiotics for human consumption when it's needed and not overuse it in dogs and cats. And it has been shown in European studies that gastrointestinal disorders often are overused with unnecessary antibiotics, just like the first case of Jycke that I showed you to start with. And I should also add that with Jycke, I started treating him when he was 7 and he's now 13 and a half, and I've never had to give him any antibiotics at any time point. So that's all for this, and I'm happy to take any question, and thanks to Perina for inviting me to lecture. Thank you.
Our next speaker is Dr. Gail Charneski-Molden. She's a senior principal nutritionist at Nestle Purina and an adjunct professor of veterinary clinical medicine at the University of Illinois. She received her bachelor degree from Cornell University and master and PhD in animal nutrition from the University of Illinois. She served multiple scientific boards and multiple scientific societies during past years. Gail has published over 60 articles and abstracts in the area of pet nutrition. Her research focuses on probiotics, prebiotics, and the effect of nutrition on gastrointestinal health and microbiome. Thank you very much, Gail. We are looking forward to your lecture.
Hi, I'd like to thank the organizers for inviting me to speak at this forum. Today I'll be speaking about a subject that's near and dear to my heart. I've been a nutritionist at Nestle Purina for 30 years now, and for the last 20 years I've been working on probiotics and symbiotics and prebiotics. And what we found is there's a lot of misinformation out in the literature and in the world about probiotics particularly. So one of the things that we're going to do is to talk about how we might select a new probiotic strain, as well as symbiotics, and some of the misinformation that's out there about probiotics in general. So for the outline of my talk, we'll be going into factors that go into choosing an effective probiotic, the strain, the species of animal or that it was isolated from, stability of the probiotic, the proper dose. We'll talk a little bit about multi-strain products versus single strains, and then we'll end up talking a little bit about prebiotics and symbiotics. So the first thing we need to keep in mind is what is a probiotic? And the definition by the World Health Organization is that probiotics are live microorganisms which, when administered in adequate amounts, deliver health benefit for the host. And there are a couple key parts to this statement. Number one, they have to be alive. That's not to say that a dead microorganism might not work, but in order to be called a probiotic, it needs to be alive. Probiotics are not a generic type of thing. All probiotics are not the same as each other. The benefits are strain dependent. And by strain, I mean not species dependent. So if you have a Bifidobacterium longum, one strain of Bifidobacterium longum could have very different benefits than another strain. Stability is also dependent upon the strain. So some strains of bacteria are very stable, some are not as stable. And this is some work that was done by Ruth Noor and Rainer Simmering a number of years ago. And they were screening different strains of probiotics that were, these were particular ones were isolated from dogs. And in Ruth Noor's work, she had looked at 75 different strains of canine lactobacilli. Of those 75, only 16 actually had potential for probiotic activity. And in Rainer Simmering's work, he saw only 2 out of the 50 having potential. So by having potential for probiotic activity, what we mean is that they had potential benefits in vitro screening systems. And there was also screening to look at whether or not they might survive through the GI tract of the pet and whether or not they would be stable enough in the environment, the outside environment, in order to potentially be commercialized at some point. So some of the strains did not have potential benefits. Some of them were looked like they had benefits, but they were dying off in the in vitro GI system, and others were fine on those two, but they had to be refrigerated, or they might only have a shelf life of a week or something like that. So all of these things together need to be taken into consideration by the manufacturer when the probiotic strain is being selected. So some examples of types of information that might be available or should be available when you're selecting your probiotic strain. The first one that always should be available is a whole genome sequence. And this is very important for looking at safety of the probiotic. There are a number of things that are looked at depending on the species of bacteria, but we'll look for genes for antibiotic resistance and make sure that they're not atypical of the species of probiotic. You'll look for potential pathogenic factors, just anything that's that's unusual about that strain to make sure that it is safe to feed. You also look at potential to produce benefits. So again, this is dependent on what the benefit is that you're looking for. You might be looking for specific enzymes that are being produced by the bacteria. You might be looking for production of certain vitamins, neurotransmitters, amino acids, other bioactive proteins. So initially, this screening could be done in silico, just looking at the whole genome sequence and looking at what your targeted benefits are and whether that strain might produce those benefits. Another thing that can be looked at, again, typically this is done in vitro to start with, is antipathogenic activity. So some strains of bacteria will produce bacteriocins, and is this important to you? And are the proper bacteriocins being produced for that strain and for the end benefit you're looking at? You can also look at in vitro and in vivo effects on pathogen growth. And another one that we typically will always look at is the anti-inflammatory profile. So what is the cytokine production of this strain of bacteria? Is it an anti-inflammatory strain? Is it an immune stimulatory strain? So basically what you want to do is to evaluate the strains against the targeted benefit that you are going for. So the most important thing about choosing strains, or another really, really important thing about choosing strains, is to look at the targeted benefit of that strain. And I think that one important thing to keep in mind is that there is no single probiotic that can do everything. No probiotic is perfect. You can't have anti-inflammatory effects at the same time that you're having immune stimulating effects, for example. So when you're choosing a strain, you need to be very specific about what benefit do you really want this strain to confer to the pet. One example of this is, again, Enterococcus faecium SF68. The graph down at the lower left shows some information on canine distemper virus and antibody levels after vaccination. So this particular probiotic was fed to puppies from 8 weeks of age up to 1 year of age. And you can see there was a very nice antibody response to the vaccine in both the control and in the test puppies. But what started happening over time was that the antibody titers for the control puppies started to drop. So at 9 months and a year of age, you can see there was a significant difference in antibody levels between those two. And this is what we would expect from an immune stimulating probiotic. We expect some stimulation of the immune system, better antibody responses, more prolonged antibody responses to the vaccines. Because of this, it's not surprising then to see some effect on fecal quality during growth as well. And this is actually the graph over on the right is from a different study. And this study, again, was done with growing puppies. These particular puppies received the probiotic from 3 weeks of age. So as soon as they were starting to consume solid food up to a year of age. And you can see that what the graph represents is the fecal score of the puppies. The top line is the puppies who were on the probiotic, and the line below that, the yellow line, are the control puppies. On this particular scale, 100 means ideal feces all the time, and the lower it is on the scale, the softer the feces were and the closer to not being firm. So what you can see is that with the puppies who were on the probiotic, their overall fecal quality was higher, and there was also less, statistically less variation day to day. And again, this is something that we would expect from an immune stimulating probiotic. Excuse me. As I mentioned, you can also look at antipathogenic effects of probiotics. And a lot of times what we'll do is we'll do screening of the probiotic in an in vitro system, culturing it with the pathogen of interest. But you can also screen for potential effects on diarrhea. And again, this is something you need to be very specific about as far as what type of diarrhea are you interested in targeting. Are you interested in acute diarrhea that might be related to a pathogen? Are you interested in chronic inflammatory diarrhea? Because this could be a very different probiotic than one that's effective versus acute diarrheas. Are you interested in a probiotic that's effective against antibiotic-associated diarrhea? You can also look at other benefits, for example, behavior, disease, performance in livestock. Often we look at growth rate, we look at effects on fecal quality from day to day. And this is an example of Bifidobacterium longum, BL999. And this particular probiotic, the targeted benefit that we were most interested in was behavior and decreasing anxiety during stressful events in normal dogs, and also the ability to decrease anxiety overall in dogs who tend to be a little bit more anxious. And so again, we screened this probiotic in vitro. We looked at the whole genome sequence. We were looking for potential to affect neurotransmitters, to affect vagal response to stressors. And the results that you see in the graphs over on the lower right-hand corner show that there was, the lighter bar is the dogs who were on the B longum BL999, and you can see that there were significant decreases in cortisol levels during a stress, significant decreases in heart rate, as well as a significant increase in heart rate variability. And heart rate variability tends to be lower when people or pets are stressed. This is when you go into fight or flight mode and your body becomes very steady, getting ready for a threat. So an increase in heart rate variability is actually a sign of a better mental state and less anxiety. So there are a number of factors that can affect the efficacy of a probiotic strain. So we've been mentioning choosing a strain that has the benefits that you want, but there are also factors that will affect whether or not those benefits are expressed. And I think one thing that most people do not know about, unless you're very intimately involved in the production of probiotics, are the production aspects of this. So one question is, if you take a strain of a probiotic and produce it with different manufacturers under different conditions, will it have the same efficacy? And the simple answer to that is probably not. If you were to buy someone else's probiotic, take it, isolate the strains out of it and grow it up, it wouldn't necessarily mean that it's going to do the same thing. A good manufacturer is going to have a really tight quality system in place. This will ensure the absence of pathogens being introduced into the system, but also consistent genetics. A good manufacturer will have a master strain or a master lot of the strain, and then working seed lots that will come out of that. The master lot is kept separately, and what we'll do over time is you prove the identity of the production batches of the probiotic and compare those to the deposited strain. We need to make sure that the strain has not drifted at all as far as its whole genome sequence goes. And that will help to ensure that the strain is still beneficial and has the same stability that it had. The other thing is that production conditions will also affect the ability of the strain to have beneficial effects. And I've seen situations where you had a very beneficial strain that was having really nice physiological responses. And when the production system was changed, the fermentation media, or at what point the strain was harvested, or how long the fermentation was allowed to go on, you could actually have a total loss of all activity of the strain. So it's important to know how exactly to produce the strain, to make sure the system is well controlled, to make sure that the fermentation is always the same, and that verify the viability of the strain in those production conditions. There are a number of publications on this as well. Some of the things that can be affected by the processing or the production methods are the actual components of the cell. The structure of the cell can change. There's some publications showing that changes in the cell structure can actually affect the efficacy of the strain, and how you're fermenting it can actually affect the actual structure of the cell. Gene expression can be affected by what's in the fermentation media, and also functionality, as we mentioned. So the pH of the media is important, how long it's kept at different pHs, the phase of growth when it's harvested, as well as the composition of the growth media. And I provided some example references for you here. There's quite a few additional publications on this as well. So the next question is, does it make a difference where the probiotic is actually harvested from? And we'll often hear people say, you know, if you're feeding a probiotic to a dog, the probiotic has to have originally come from a dog. Or if it's going to a human, it needs to have come from a human. The question I have is, where's the proof of that? When we look at colonization, so the argument that's typically made is that if you have a species of bacteria that's native to an animal, it's going to be able to colonize better and survive better in the GI tract. For most probiotics, we're actually looking for transient colonization, not permanent colonization. We want the probiotic to be able to go away when we stop feeding it, because it might not be necessary anymore, or it might be doing some things that you want it to do short term, but not necessarily want to have that probiotic live in the animal forever. So having transient colonization is fine. So that basically means when you're giving the probiotic, it's alive in the system, it will survive in the pet's gut, and it can do what it needs to do. But when you stop feeding it, within a couple of days, it'll probably go away. Probiotic strains can be perfectly efficacious with that type of colonization. So the other thing to keep in mind is whether or not your target is a target that would be present in all species. There might be certain benefits related to the physiology of the host that might only be relevant for one species of animal. But if you're looking at more general benefits, or benefits that would have a broader physiological activity across different species, then having a in-species strain, or a strain that was isolated from a specific host, is not as important. So again, some examples of this is, I'll use Enterococcus faecium SF68 because I'm most familiar with this probiotic. This is a lactic acid bacteria that has inhibitory effects against enteropathogens. For example, Salmonella, E. coli, Shigella. These are enteropathogens that can live in virtually almost most mammals could potentially have these. And this particular probiotic will kill these, or have antipathogenic effects against these bacteria, regardless of what environment they're living in. This is also an immune stimulating probiotic, and again, will have this effect across multiple species of animals. It was originally isolated from the feces of a human infant, but there are numerous publications showing efficacy in humans, for example, before, during, and during antibiotic therapy in people as far as preventing traveler's diarrhea. There's a host of publications on livestock, different types of livestock, pigs, poultry, cattle, rabbits. I've seen information in zoo animals as well, having antipathogenic effects, growth promotive effects, effects on fecal quality. And there's quite a bit of data in pets as well, showing effects on fecal quality and microbiota stabilization. Personally, looking through the literature, I think this is fairly typical that when you have an effective probiotic, that'll be effective across a variety of species of animals. I'm not sure actually I've ever seen a probiotic that only works in one animal and only works in the animal it was isolated from. It's also important when you're selecting a strain that you want to maintain the viability of the probiotic. As we mentioned earlier, the definition of probiotic includes that they are live microorganisms. And there are a number of factors that can affect viability of probiotics. Oxygen, exposure to oxygen is very important, or lack of exposure to oxygen. The food matrix that the probiotic is present in makes a difference. The water activity or moisture present in the product, typically at a higher water activity, you will start seeing a loss of viability of the probiotics. Basically, when we have probiotics in a matrix, we want to have them kind of in a state of suspended animation. You don't want them actively fermenting unless you're in a situation, for example, if you're giving a yogurt to a pet or to a person, they would be alive at that point and active. And but those products only have a shelf life of a few days. The temperature is also very important. Typically, if you go too high, you're going to start seeing some loss of viability. The amount of time from when the probiotic is produced is very important, as well as the strain. So you can have different strains of the same species who have different viability. And when you start mixing these conditions together, you also will see a more rapid drop in viability. So for example, if you have a high temperature in the presence of high moisture, the probiotic will die off quicker. So it's very important to make sure that you're getting your probiotic from a manufacturer who is looking at the viability of the probiotic to make sure that the probiotic is still alive at the time it's fed to the animal. Okay, it's also another thing that I've heard a lot is, in order to be efficacious, your probiotic you have to be feeding 1 billion or 10 to the 9th bacteria per day. So is this true? Do you really have to have 10 to the 9th bacteria per day? And if you have a probiotic on the market that has fewer bacteria, does that automatically mean it's not going to be as effective? There have been very, very few dose response studies published on any probiotics. Most of the probiotics on the market that are efficacious, the efficacy studies were done with 1 billion bacteria per day. And if there was dose response work, it was not published on it. So basically the manufacturer would pick a dose that they thought would work. And I could tell you, we've done this as well. You feed that. If it works, then you might just go ahead and launch the product with that level of bacteria and not go back and look at whether or not a lower dose would actually work. So again, here's an example with Enterococcus faecium SF68. This is a kitten trial, and the probiotic was fed to these kittens from weaning to 1 year of age. And we had either control product, 5 times 10 to the 7th CFUs bacteria per day, 1 times 10 to the 8th, or 5 times 10 to the 8th. In this particular study, there was an outbreak of diarrhea, and the incidence of diarrhea was lower in the kittens who were fed SF68 than in the control. And these kittens also recovered much quicker than the ones who did get sick recovered much quicker. What we saw when we looked at the doses of the probiotic is that the 5 times 10 to the 7th was just as effective as the 5 times 10 to the 8th. And the product we have in the market has 5 times 10 to the 8th. This gives us a little bit of leeway in case there is some loss of viability. But again, you can see this is quite a bit lower than the 10 to the 9th bacteria that often you'll hear have to be present in a probiotic. So, basically the important thing is to make sure that whatever dose is in the probiotic, that's the dose that the efficacy studies were done at. Another thing that you'll often hear is that multi-strain probiotics are better than single strains. Having a variety of bacteria in the probiotic might potentially enhance efficacy. So the question is this true? Possibly. I'll say yes, no, maybe. It depends on what the bacteria are. You need to look at the targeted benefit, and the strains that are in the product need to be picked based off of what they do in the system with the other probiotics, but also how they might affect that benefit. So if you just randomly say, okay, I'm going to take 3 bifidobacteria, lactobacilli, and an Enterococcus, put them together, that's going to be better than any of those probiotics alone. This is not necessarily true. So if you're looking at, for example, an immune benefit, if you take an anti-inflammatory strain and mix it with an immune stimulatory strain, what effect on the immune system are you going to get? You can't have immune stimulatory effects at the same time as anti-inflammatory. One of those will predominate. So which benefit are you looking for, and what happens when you mix these strains together? What does the data say about this? Are there actually studies looking at efficacy of multi-strain products versus those individual strains to make sure that they actually work better in combination than alone. And there's very, very few publications that are actually comparing the probiotic to its individual component strains. There are some publications that show efficacy of multi-strain probiotics versus a single strain, but the single strain is not a strain that was in the multi-strain product. So it could be that the multi-strain might be better than that single strain for that particular use, but it doesn't mean that it's necessarily better because it has more than one bacteria in it. So as I said, there's few that are comparing multi versus single strain. There's even fewer publications that compare a multi-strain product to its component strains. Here's a couple examples of these. In the first publication, basically none of the combinations, they looked at a couple of probiotic bacteria and looked at combining the bacteria and found that none of the combinations had additive or synergistic effects on the cytokine profiles that they were targeting. And actually the cytokine response of the mixed product was actually closer to an average of the responses of the individual strains, which is really what we would expect to happen in most cases. And so by having the combination of bacteria, they were actually blunting the effect on cytokines. So there was a really nice review article that was just published, and the conclusion really was that multi-strain probiotics might be more effective than individual component strains if the effects are synergistic, but there's really a lack of research in this area. And I know when we've looked at multi-strain products, we have some under development right now. We've selected for strains that have very specific benefits and have in vitro data showing, or in vitro data showing that the combination of the strains actually is better than the single strain, than each of those strains alone. So it really just depends on what the product is. You can't make a blanket statement that multi-strains are always better than single strains. So when we look at the product label for probiotics, there are a couple things that we can get a hint from as far as what's on the label. First of all, the ingredient statement should include the specific probiotic or probiotics that are present. There should be a guarantee of live bacteria, and that guarantee should be at the end of shelf life. Occasionally, you'll see a product that says guaranteed at the time, it has this number of bacteria at the time of manufacture. That's always a red flag to me, because we're not feeding the probiotic in the factory where it's being made. We're feeding it maybe months after that. So we need to make sure it's alive at the end of shelf life. If it's a blend of probiotics, the guarantee should be for each genus. So I'd like to finish up just talking a little bit about prebiotics and symbiotics. So as we mentioned, probiotics are beneficial bacteria that are fed to the animal, whereas prebiotics are food for the beneficial bacteria that are already in the gut. So in order to be considered a prebiotic, it needs to be a non-digestible food ingredient that is beneficially affects the host. And typically these are broken down by the bacteria that are present in the gut. They're resistant to digestion in the upper GI tract so that they can actually be food for the bacteria lower down in the GI tract. And they provide a physiological benefit. I think the thing to keep in mind here, because there's some confusion about what is a fiber versus what is a prebiotic. All prebiotics are fibers, but not all fibers are prebiotics. A symbiotic, on the other hand, is a mixture of more organisms together with a fiber or a prebiotic or another compound that has an effect on the host. There can be a complementary product, so you have different ingredients that have different benefits, but they don't interact codependently, or you can have a true synergistic effect where a substrate that's in there or the prebiotic is actually used by the co-administered probiotic. And either of these situations can be true. So just in conclusion, when we're looking at development of probiotics and symbiotics, we need to have a very defined target. The strain of bacteria and or fibers should be chosen based on that specific target. The production of the bacteria should be optimized to optimize efficacy as well as stability, and how these bacteria are produced does make a difference on both of these. There should be efficacy studies done with that specific strain of the probiotic, and the dose should be based off of efficacy studies, and higher is not necessarily better. You just need to make sure that it works at the dose that it's being fed at, and that that dose is still present in the final product. So thank you for your attention, and we'll be having a question session at the end of the set of presentations.
Our next speaker is Dr. Mike Lapin. Dr. Lapin graduated from Oklahoma State University, completed an internship, internal medicine residency, and PhD program in parasitology at the University of Georgia. Dr. Lapin is the Kenneth Smith Professor in Small Animal Clinical Veterinary Medicine at Colorado State University. He is the director of the Center for Companion Animal Studies, and he helps direct the Shelter Medicine Program. He is the chair of the Wasawa One Health Committee. His principal areas of interest are prevention of infectious diseases. His research group has published over 300 primary papers or book chapters concerning small animal infectious diseases. Dr. Lapin received multiple national and international awards for teaching, outstanding contribution to feline medicine, scientific achievements, and clinical research. Dr. Lapin, we are looking forward for your presentation.
Hello, everyone. This is Mike Lapin at Colorado State University, sitting in my office in beautiful Fort Collins, Colorado, wishing that we were all together in person. As always, please follow up with me on my CSU email address listed here. I'm more than happy to follow up with questions about this talk. As many of you know, I'm an infectious disease interested person with my diplomate status being in internal medicine. My PhD is in parasitology, which got me interested a little bit in our diarrhea slide. And I look forward to sharing what we've learned in our group and what I've learned from the literature about some of the ways that we might consider selecting probiotics and symbiotics that we might use in our veterinary practices. I do get a chance to work with many of the different great pet food companies, as well as drug and vaccine and EPA product companies. But today, working with the Institute, which I'm very proud to be part of, we'll only talk about any product-related things if there's a peer-reviewed publication. My definition for my talks would be at least at the level of an ACVIM abstract, where other peers reviewed that work. I think we would all agree on this slide that the labels are correctly placed. The sooner that we can get a stool to go from a score, and using the Nestle Purina score, this would be kind of a 7, that watery part, to get that back to a nice score of 3 as soon as possible is really our goal with diarrhea cases, because as we improve the diarrhea, we also lessen the risk of potential transmission of zoonotic diseases. And so you'll have a great talk from Gail at this meeting as well, a true microbiologist that designs and selects probiotics. And so we've worked together to try to strengthen each other's talks, but not replicate. So we'll be using these definitions throughout my talk, and in Gail's talk, you will have seen how they actually are selected and proven dose-wise. We'll both be emphasizing some of our issues with what we know and what we don't know over the course of those lectures. So I wanted to give a shout out to Dr. Schmidt as well as Elena. These 2 review papers that have come out in the last 4 years are really great to have in our access to what we've learned about this field. And it's amazing. Today we'll learn many, many things, and tomorrow as well, about advances since these reviews came out. But it is amazing how far behind in small animal world we are compared to humans. Again, we barely have enough data to do a single meta-analysis on any of the topics that I'll talk about today. So what I've been speaking with veterinarians and other specialists working in this space is that there's pretty good data when it comes to the GI side of things, and we'll talk about some of those hallmark papers in this 30-minute section. I would, in this section, also like to talk about effect on anxiety. We'll have a few words about atopy as well, and how probiotics might ultimately choose to be beneficial. But I also wanted to talk in the second lecture today about some of the immune modulation things that are now proven with some of the bacteria that are used in probiotics and symbiotics. One of the things that Gail will have emphasized in her talk is that you've got to have live organisms to be a probiotic. And so I think all of us that lecture in this space use both of Scott's papers, Dr. Weiss's papers, where they just merely selected products claiming or purported to have probiotics, live organisms, and then showed just with routine culture techniques that we couldn't even document the appropriate organism in some probiotic products, as well as bacteria that you might not have wanted to have in that probiotic. And so my first hint to those of you that do this for clinical reasons, you really should work with a company, and preferably veterinary company, in my opinion, that actually can show you their stability data. And really what you want to know when you ask that question is you want to know what are the numbers of bacteria at that end of the shelf life. And so that would be a key question I would ask anybody, and I'd want to see in manuscripts. And then, the second level of evidence that I really like to see from a company product that I'm going to purchase to use in my clinic, I really would like to see some sort of project published. And so today we'll share a number of those as we go through our time together. But I'm perfectly accepting of data on file if they can actually show you that information, proving that that probiotic or symbiotic actually works for something they're claiming on their label. And why we have to ask that is that, it's just not as tightly regulated as FDA drug products or EPA insecticides. So again, what I recommend to my vet students, my interns, my residents, and our pharmacy is get at least those two data points: numbers of organisms at the end of shelf life and some sort of data published. And that data could even be on Venn. If there's a group of people that have posted on Venn that a probiotic that you might not know much about seemed to work, that's a level of evidence, right? But getting it into a peer-reviewed publication is just a little bit more high-powered. Now, Gail has already discussed this, or will discuss this with you, so I'll keep it short today. But again, not all companies can tell you how they determined their optimal dose of bacteria. And then, of course, there are many products that have more than one bacterial organism and even yeast organisms that, what happens when you use them at the same time? Yeah, what if you had an immune enhancing and an immune dampening probiotic in the same mixture? You might actually be counterbalancing. And so I'm always curious about if you had 4 or 5 organisms, 6 organisms, how did you actually prove that those organisms correlate in a positive way? The bottom line is, even though we don't often know how they came up with those decisions, what we do have now, or we continue to ask for, are actually papers showing that product as designed actually works. And so that's what I'd like to do with you now for the next, 24 minutes or so, is kind of talk about, the evidence-based things. It'll emphasize some of those points today. And so I think clinical response data, we actually have probably the strongest data that actually, if you had a case like a diarrhea case, that you used a certain probiotic or symbiotic and showed a treatment response either faster than not treating, or in a peer-reviewed paper with some crossover design. Yeah, the clinical response data is pretty convincing in a number of these studies that I'll show you. Teasing out how they have their effect has actually been a little bit problematic. So I hope to learn a lot over the next 2 days from Jorg and Jan and others, talking about what they've learned about how to actually use these assays to assess the metabolome and the microbiome a little bit better, to explain why we see some clinical response things. So I wanted to show you Audra's paper first. This is one where we were quite interested in looking at the Fortiflora, one of the commercial products that contains Enterococcus faecium SF68. We wanted to get a better feel for, you know, these things cost about $1, $1.50 a day. We wanted to see if that would be worth it, you know, to say a shelter to use this product with their diarrhea cases. And so if you remember this one of the first things that we did was show that in this antibiotic probiotic combination, you could give them at the same time. Yeah, we showed that the SF68 strain of Enterococcus faecium was resistant to metronidazole. So in this 32-dog pilot study, we did actually give them both the probiotic and the metronidazole at basically the same time. Obviously, in this study, as you can see, there's a placebo, which is the probiotic without the organism, so everything that's in the commercial product, but no bacterium. We do a lot of our studies as pilots with approval from our clinical review board or from our IACUC. And then if we get statistically different data, we might go on to paper publication like we did with this study, or we might use the data to develop a power calculation to extend the study. Well, this one, I just wanted to remind you, the shelter uses metronidazole for all their nonspecific diarrheas. And as we'll be talking through this lecture, we don't do that anymore, many of us, but that was what they currently did at the shelter. Thus, we were looking to see if we could speed resolution of diarrhea or give a consistently better stool by adding the probiotic. So the one statistical punchline from that paper was that if we looked at the percentage of normal stools, we actually did show a statistical difference if we gave the probiotic containing with the bacteria versus the placebo. And so it's one of those first papers, I think, in veterinary medicine with a commercial product that showed that using that probiotic on top of an antibiotic would potentially be beneficial. About that same time, a little bit later, we were actually looking at, could we lessen rates of diarrhea in shelter animals? And in this particular study, we actually showed that we could lessen diarrhea rates in cats. Notice the title says dogs, but we actually couldn't show a difference between dogs using the probiotic versus the placebo, because in the dogs, when we put the whole shelter on a standardized diet, the dogs drop from about a 15% diarrhea incidence for 2 days or longer down to about 3%, just a good standardized diet. However, the cats, as you can see from this summary slide, they still had diarrhea even on a standardized diet. And so here the placebo are the closed bars, the probiotic supplemented at times are the open bars. And what we were doing in this one is really looking at a effect on a room. So we fed the probiotic for a month, the placebo to the other room for a month. And then to account for room effect, we crossed them over for the second month. And as you can see, there was a statistical difference in the kitties that were supplemented in the stray room, a numerical difference when they were supplemented versus placebo in the feral room. And then that statistical significance did persist after we combined the data. So again, it's pretty good evidence that in a stressed environment with a lot of infectious diseases, co-infections, that actually use of a probiotic could further drop feline diarrhea rates. So again, showing a clinical effect. This was actually with a nice symbiotic produced by our friends at Nutramax. Craig did this one, Dr. Craig Webb here at CSU. And this was an interesting one because, you know, we have those kind of chronic diarrhea cats, so we can't really figure out what's wrong with them. So this was an open trial, and they showed both improving fecal scores over time with this particular probiotic, or excuse me, symbiotic. And they also showed a really high owner satisfaction rate. The owners felt there was a positive response. There's some similar data with some of the cats housed in the Purina facilities, the taste testers as well. So basically, we have some data suggesting that commercially available probiotics can lessen acute or chronic diarrhea rates in cats with nonspecific diarrhea. So what we did at our group, we then became very interested in seeing if we could start gathering more data about the effect of antibiotics and probiotics, specifically starting with, could we lessen antibiotic diarrhea by co-treating with a probiotic? And then later on, we expanded to looking at treating diarrhea that has already occurred with a symbiotic. So I'd like to share those with you next. But bottom line is there's tons of data in people. It's a little bit all over the place, but the overall assessment is that appropriate probiotics, and it wouldn't be all of them because they all behave differently, but pretty good evidence that probiotics can lessen antibiotic-associated diarrhea, again, because those antibiotics are often very hard on the GI microbiome. So this one was done by Camille Torres Henderson, who's our current nutritionist here at CSU in our group. So we selected cats from our research facility. We housed them in cages and kicked out any of those cats that were kind of predisposed to having stress diarrhea. And then if you don't remember this particular paper, many of the human papers give the probiotic first, followed by the antibiotic. So we chose to go with that design. We also showed that in this study, calavulanic acid actually kills this probiotic. So in contrast to metronidazole not killing the SF68 strain of Enterococcus faecium, in contrast to metronidazole, where you can give them at the same time, this particular study, we separated them. And I kind of like the theory about giving the probiotic first instead of the antibiotic first, with the thought being you might as well give the probiotic, let it start doing its thing before you give the antibiotic that will likely kill it. And so, again, this is a published paper already, so I don't want to belabor this for too long. But when we applied the Nestle Purina scoring system with a 7 being water, we actually found that we saw a 6 or a 7 in most of the cats, with or without the probiotic, without the bacterium. So take-home message is clavulanic acid in cats causes diarrhea like crazy. And if you were thinking it might be a little bit lower than 70 some odd percent or higher, it's probably because they were having like a 5 and the owner didn't notice it because of clumping litter. The point is there were actually no differences if we just looked at 6s or 7s between the groups. But when you break down those distributions a little bit more granularly, you'll see here that when we looked at the placebo group, only 7s in the placebo group. Fortiflora had no 7s. So that was the only statistically significant difference amongst these numerical differences. But the numerical trends were towards a positive effect. And then if we look at the entire 10-day study, then basically the cumulative diarrhea scores, the how many days you had a bad stool over the course of the study was significantly improved in the group that got the live bacteria, not just the placebo. So again, those kind of studies are beneficial. We just have so many unanswered questions like, should you give the probiotic first or not? All we can say at this point is that's what we did. But interestingly, and it'll be great to hear Dr. Jan's talks over the course of this particular webinar series, it's really tough sometimes to see why they were getting better. So we had the clinical responses, but the microbiome responses were a little bit tough to figure out exactly how it was helping them. And then if you look at the great data from Jackie Whittemore's team using symbiotic from our friends at Nutramax, it's really interesting to see that we have effects, clinical effects, less hyporexia, less vomiting, but maybe lesser effect on some other signs. And then even when you do really, really elegant work with the metabolome and the microbiome, trying to sort out exactly why those clinical effects are recognized is a little bit tough. But I think if you contrast the clindamycin work with the clavamox work, I think we would all agree that there's probably differences between products. So we need more head-to-head to help us decide what probiotic might be best. Remember, you can't say best unless they're compared. So once the Purina team added psyllium to their SF68 bacterium, so this is now the Fortiflora SA product, we decided to, in this study, go ahead and induce or give cats clavulanic acid. And then if they develop diarrhea, we went ahead and took all the cats off clavulanic acid when their diarrhea started. And then half of them got the probiotic. The other just had the antibiotic removed. And I did say probiotic, but this is a symbiotic. So point is that once we did that work, it was pretty amazing. If you look then at the cumulative scores between the two, so greater than 5 on the fecal scores, there was a significant difference. So this one was, again, a paper that looked at, you might be able to treat diarrhea with some probiotics if it was induced by the appropriate bacterium. So again, I think overall we would probably all agree that we've gotten some data with at least the beta-lactams, with clindamycin, with at least 2 probiotics that are commercially available, and that perhaps using those probiotics with those antibiotics could be beneficial. Are we ready for a meta-analysis? No. So I think we're ready to continue to get more data. So here's a nice one. I'll just give you a brief blurb on this one. Jan and I were chatting about, should we be saying for Giardia, always start with a benzimidazole rather than metronidazole, ronidazole, or secnidazole, because we knew the antibiotic anti-Giardia drugs were really messing up the microbiome. But, neither one of us at the time knew whether or not the benzimidazoles, Avantel or fenbendazole, we actually didn't know if they affected the microbiome. So Madeline Fukuhiro did this particular paper, team approach between our friends at Texas A&M and here at Colorado. And good news is benzimidazoles don't seem to affect the microbiome. And so most of us now do recommend, if you're treating Giardia, you should start with the benzimidazole, and you probably don't need to use it at the same time with metronidazole. Say metronidazole as your kind of rescue. Those of you that treat immune diseases, either gastrointestinal or systemic, might be interested in what we just showed with mycophenolate. Yeah, mycophenolate diarrhea, what's about 25% when we use the 10 mg per kg twice daily. Well, this particular work, we were able to use, and these are fecal scores over time, we were actually able to show pretty soft stools with mycophenolate in some of these animals over time. And here's when we then did a group that were treated with the SF68 symbiotic, the one with psyllium. Yeah, you might have seen that last year at the internal medicine meeting. So so then using a probiotic or a symbiotic, I'd probably use the ones that were published for that purpose to lessen diarrhea due to antibiotics, vomiting, and or diarrhea due to an immunosuppressive drug are probably a good idea. We're also in that era, I think now, is that most of us won't use hyalocin up front. Yeah, I think now because we know that Clostridiums and Salmonellus, Campylobacters, enterotoxigenic E. coli, frequently they're going to respond to the appropriate diet and the probiotic or symbiotic. They probably don't need to have that antibiotic up front because we now know those chronic effects on microbiome might be negative. So I do urge practitioners in the space to go ahead and think about doing the probiotic diet change first. Hey, this is a really nice paper I wanted to share with you guys today, just pointing out that, the fiber, I think we're always thinking about with large bowel, that it's also that prebiotic effect that might help our probiotics actually do a great job. So a neat paper supporting the use of appropriate diets with probiotics, if they're effective for that purpose, could lessen some of our diarrhea rates. The trichomonas story, as you guys know, going in and telling an owner that good news, the diarrhea may be gone in 9 months, it's not a very happy time. And kudos to Dr. Gukin with all the efficacy work, and then Dr. Mark's team showing the first neurotoxic cases, because this one crosses the blood-brain barrier even better than metronidazole. The good news is that we have at least one drug. The bad news is, and here's a great paper out of A&M. The bad news is they don't all respond to ronidazole. Now, one of the great papers that then just came out here in the last 2 years was looking at those Enterococcus faecium and could they be associated with, inhibition of T fetus growth? And the answer is yes. And so very interesting, and might be great evidence to help you if you have a little pixie bob cat with tritric that's not responding. You might think about this probiotic in the Enterococcus faecium group and retreating. So this was a paper that a probiotic was part of the pathway. This was also Enterococcus faecium SF68, where it didn't show an increase in survival. A little bit hard to tease out the diarrhea part of the study, but as you can tell, when you do even really a pretty good number of animals, it's always a little bit hard to tease out the efficacy data for these things. Again, the vitamin supplements seem to be associated with increased survival, but not many of these other things. But then, of course, the increased risk for death with sub-Q fluids and antibiotic use, doesn't necessarily make sense either. And so that's probably could be related to that's just the sicker animals that got those treatments. But bottom line is we're not going to always see a positive effect of antibiotics with or without probiotics in some of these cases, which does lead some of us to say in our talks, you might want to keep a second probiotic ready. Hey, we're looking forward to getting more commercially available probiotics or symbiotic data for constipation. And I look forward to seeing you over the next year, because we just started 2 different studies that I can't tell you about today that we'll have a little bit more information about. Could these products perhaps help diarrhea or constipation? So stay tuned for that. And then the last little blurb I wanted to share with you today in this 30-minute segment was use of bacteria for other things. Again, we'll talk a little bit more about atopy and the immune lectures, immune stimulation, immune dampening in my next lecture. But I thought in this particular topic, it would be a great time to remind you about the Bifidobacterium longum group, known in a number of human studies to change some behavioral things, especially in folks with inflammatory bowel disease, changing some of that anxiety and things that might be associated with the gut-brain axis. So Reagan's work that was published as a proceeding, coming as a paper in the near future, that led to use of this strain, the Bifidobacterium longum, strain 999, to use that to lessen anxiety in the dogs, actually did lead to a commercial product called Calming Care. So that was really great work. And I've got to admit, I've been negotiating with our friends at Purina for a couple of years about perhaps using this bacterium, or trying to find a model to use this bacterium to lessen anxiety in cats, for lessening stress in cats. And so I can't show you the full day-by-day data, the whole design, because this one is currently in review, but I can show you then one of our peer-reviewed abstracts. So basically, this strain, Bifidobacterium longum, strain 999, is exclusive to the Purina team, already in a commercial dog product. Now, it's recently been launched in a feline product as well, that I will direct you in a to a non-commercial or a commercial event to talk about the product itself. But the label that came out with the cat product was largely tied in with this study. So short version is we took cats with chronic herpes, came out of one of my vaccine studies, they're in remission. We went ahead and then fed the probiotic versus the placebo for 42 days. Again, we don't know exactly when the maximal effect is, so we chose to do the equilibration period for that 42 days that we think is the maximum time required in a dog. Then, to induce stress, these cats are in their two different group house rooms. We then changed them from group housing into cages, back into group housing, back into cages, while following a number of herpes type things, the clinical things, fever, et cetera, cortisols, as well as some of the behavioral parameters. And so in the short version of this particular talk today, I wanted to point out specifically that the cats enjoyed eating the product, either the placebo or the probiotic. We did show evidence for less stress during those stress changing periods by cortisol concentration. We believe that the less sneezing in the probiotic group was because of less stress, less reactivation of herpes. And then we found a number of things that suggested these cats had positive effects on their behavior. Reaching out, asking for attention, less pacing when they're on the probiotic were two of the major ones that we were able to find. And we also found that these kitties were less likely to have elevated body temperature over time. Really appears that there could be an effect on anxiety and stress in cats with this probiotic, and even lessen a physical disease, the reactivation of herpes. So continued studies with this particular bacterium and this probiotic going on both in field studies and in research studies. But I thought you'd be interested to hear about it. And again, it is in a product now that's purchasable in the United States and soon probably in other countries. So thank you so much for listening to me in this 30-minute segment. Please, if you have any questions on that particular lecture, give me an email at mlappin@colostate.edu, and I'll see you in the live question session later. Thank you very much.
I have a pleasure to introduce our next speaker, Dr. Stefan Unterer. Dr. Unterer is the head of the gastroenterology services at the Clinic of Small Animal Internal Medicine of the Ludwig Maximilians University of Munich, Germany. He completed his internship and residency in small animal internal medicine at the University of Zurich, Switzerland, and University of Georgia, the USA. Stefan Unterer became board certified by the European College of Veterinary Internal Medicine in 2003. After 1 year of private practice, he returned to the academia and became a faculty member at the University of Munich. Stefan Unterer will start his new position as a professor chair of small animal internal medicine and director of the small animal internal medicine clinic at the Vet Suisse Faculty Zurich, Switzerland from February 1st next year. His clinical research project includes the intestinal microbiome, acute hemorrhagic diarrhea in dogs, intestinal barrier function, and long-term consequences of acute enteritis. Dr. Stefan, we are welcoming you for your presentation.
Hello, everybody. Thank you so much for inviting me to present at this Purina Microbiome Forum. And I'm happy to give our approach or view about the appropriate antimicrobial use in acute and chronic diarrhea. In context with this presentation, I have nothing to disclose. As you all know, bacteria are very important for every individual. And especially we have some very important beneficial effects. You know, there is a protection against pathogens of the bacteria in the intestinal environment. The intestinal microbiota is very important for the immune system development. And another example is that it is also important for the intestinal barrier function. And that's the reason why we really try to avoid to give antibiotics unreflected to every patient. But we also know that bacteria might have some harmful effects. That means a role as a pathogen. And they can cause a local damage of the intestinal mucosa. In this case, if the dog or cat is doing still fine clinically, we rarely need to give antibiotics. But sometimes we see in these cases that we have systemic inflammatory response. And in these cases, we should usually have to give antibiotics. On the other hand, we can also have a dysbiosis in context with a chronic disease, for example, with chronic diarrhea. And usually, many years ago or some years ago, we treated these patients with antibiotics on a regular basis. So nearly every dog was under a treatment trial with antibiotics. But nowadays, we think that we create also a dysbiosis by giving antibiotics. And this can maybe change the metabolic function, the immune system. And that's the reason why we reconsider not to give antibiotics in every case, for example, with chronic diarrhea. But I will explain the indication of antibiotics with some cases. And I thought this might be more interesting for you if we go through different cases and decide when we have to give antibiotics and when we can avoid antibiotics. And I will start with the first case is Maxi. And Maxi was presented with acute diarrhea. It is a Labrador Retriever, 6-month male. And in addition to the acute severe diarrhea, the dog was really depressed. So he was not happy and alert, he was depressed. And more important, we had a rectal temperature of 40.5 degrees. That means we always have to decide if this is an uncomplicated diarrhea case and if we can send the dog home with symptomatic treatment. But in this case, we really hesitated to send the dog home because he was depressed, and more important, he had this high rectal temperature, which was most likely due to true fever and was not hypothermia because the dog was not very excited, the environment was not very hot, so we thought this is true fever. So we performed a CBC in this case, and we saw that the hematocrit, the PCV, was elevated with 64%. So we underestimated maybe on clinical examination that the dog was really significantly dehydrated. So we need fluid therapy in this case. But also very important, we saw that the dog had a high number of neutrophils. So the mature neutrophilia of 25,000 per microliter really reflects that we have a systemic inflammatory response. So this cannot be due to excitement and a stress leukogram. And more specifically, we also saw that this dog had a left shift. So the band neutrophils were also elevated. So we saw, or we knew from the CBC, that this dog has a systemic inflammatory response. So the problem list in Maxi was that we have acute severe diarrhea, a systemic inflammatory response, and the dog was dehydrated. And our assessment was, we always are thinking about where might be the location of an infection if we see a patient with fever. And in this case, because this dog showed so significant diarrhea, we thought that really the intestinal tract might be this organ which has, or from which the inflammation is going on. And so the assessment was either we have a really primary infection with an enteropathogen, or we have maybe a viral infection or some other causes of acute diarrhea, but a barrier dysfunction and spreading of bacteria translocating into the systemic circulation from the GI tract. So we wanted more diagnostic to clarify this. So we performed fecal flotation. It was a young dog, but this was negative. No Giardia, no helminths. We also performed a fecal culture, and I already want to mention here that we not usually perform fecal cultures. only in the way if we really think that maybe we have a bacterial infection, that means a dog with fever, for example, and then we want to try to isolate an inciting enteropathogen from the intestinal tract. But we also performed ultrasound in this case, and we really saw that we have an area of about 10 to 15 centimeters where we have changes of the jejunum. The wall of the jejunum was thickened. We did not see a very nice layering. And you can see on the left side, we have the thickening, and on the right side, this is the same size of the ultrasound image. This is a normal layering and normal thickness of the jejunum. So if we would have an older dog, we would think this might be some neoplastic infiltration, but in this young dog, we thought this might be more an inflammatory response in this case. In addition, we saw on ultrasound that we have an enlarged lymph node, and this abdominal lymph node was not very huge, but it was very round shaped, and more specifically, we were concerned about this hypoechogenic center of this lymph node, which might reflect that we have an abscessation, that we have some fluid in the center of this lymph node. So always, if we try to culture a bacterial strain which might be responsible for an infection or inflammation, for a systemic inflammation, we try to culture an area which is normally sterile. So besides the fecal culture, we performed a fine needle aspiration of this lymph node, and we cultured the material from the fine needle aspirate. And we diagnosed actually a salmonella infection in this case. And the great thing was that we could culture salmonella from the lymph node aspirate, because otherwise, if we have a positive culture from the feces, we always do not exactly know if this is really a true salmonella infection, because we also see some salmonella sometimes in healthy dogs. But if we can culture it from the lymph nodes, we really think that salmonella spread from the intestinal tract into the lymph nodes and into the systemic circulation. And this explains the systemic inflammatory response, the fever, the neutrophilia, and left shift. So in this case, also, if we would not have been able to culture this bacterial strain, we need to give antibiotics because the dog is not feeling well, has severe diarrhea, has fever, a systemic inflammation. And in this case, especially if we have not the culture result back, we really need to give broad spectrum antibiotics. That means it does not help to give, for example, metronidazole or tylosin to these cases. We really need to cover a broad spectrum, and that's the reason why you should consider to give, for example, amoxicillin with clavulanic acid or ampicillin, and in combination with, for example, fluoroquinolone. Maybe you might be afraid of the young age, but I think to save the life of these patients, it does not make a big difference. So you can also start with fluoroquinolone for a short time in such a case. So we see that we need antibiotics in this dog with an acute inflammatory response, but we are thinking ahead what might happen after treating with antibiotics a young patient. And we know, especially extrapolated from human medicine, that a dysbiosis created by antibiotics in a young individual might be a risk factor for an immune-mediated disease, immune-mediated inflammation of the GI tract or allergies like asthma later in life. And that's the reason why we consider to give, for example, a fecal microbiota transplant to these young patients after antibiotic treatment. What we would do nowadays is we would check the dysbiosis index 2 weeks, for example, after stopping the antibiotics, only to see if we have signs of a significant dysbiosis. In this case, of course, we would really consider to give an FMT, but also if we would not perform an assessment of the microbiota, we might consider to give a FMT to these patients. Also, if they are not symptomatic. So I want to switch to another case. That's Froni. Froni has acute diarrhea, and this is a mixed breed dog, 3 years, female. And this kind of diarrhea seemed not to be very complicated. So the dog is alert, has no abdominal pain, is not dehydrated, has a normal temperature. And in this case, also we performed some diagnostics, fecal flotation, which was negative. The hematology, every parameter was in the normal limit. So we had a tentative diagnosis of acute uncomplicated enteritis in this case. And of course, we would not give antibiotics to such an uncomplicated diarrhea case. We only would change the diet. We would maybe modulate the intestinal microbiota by, for example, probiotics. Very rarely we give with this uncomplicated cases a fecal microbiota transplant without previous antibiotic treatment. You might consider to give some antiemetics and pain management if this is necessary. But I think most important is that we monitor these cases and the owner gives us feedback after a couple of days so that we are sure that this is really an uncomplicated case and not progressing to a more severe diarrhea or developing fever or systemic inflammation. And we know from several studies that we do not need to give antibiotics in uncomplicated cases. And you don't have to read the details of this slide, of this table. It only should show that we have more or less 4 important studies comparing antibiotics with placebo or probiotics in dogs with acute diarrhea. And we had a very similar outcome of all these cases in which we see that most dogs really recover within a couple of days, and nearly every dog with uncomplicated acute diarrhea is normal after about 1 week. So they typically resolve spontaneously, and we know that there is no clinical benefit to give antimicrobials in dogs with uncomplicated diarrhea. There's only one study with comparing metronidazole with placebo, and they showed that there was a faster resolution of clinical signs of diarrhea in dogs with metronidazole. But still, we think this does not justify to give metronidazole to every patient, because all dogs, also with a placebo or with a probiotic, they will be cured after at least 7 days. And we know if we give antibiotics, we can really induce a dysbiosis, and we also can produce or create the development of resistant bacteria. That means most of the time with acute diarrhea, we have some destruction of the intestinal mucosa, we have some dysbiosis in these cases, but if this is uncomplicated and we do not treat with antibiotics, normally after 1 week, we again have a nice barrier function, a nice lining of enterocytes and of this mucus layer, and a normal microbiota again. So without antibiotics, we should have a normal microbiota environment and the normal intestinal barrier in these cases. But if we give antibiotics in these cases, we know these patients might recover from the acute diarrhea, but with antibiotics, we usually create also a dysbiosis. And a prolonged dysbiosis might be a problem and cause an inflammatory environment, and it might also, of course, be responsible for the development of these resistant bacterial strains. So we are very strict in our institution with antibiotics in acute diarrhea, and I give you our criteria for justifying to give antibiotics. So first, in acute diarrhea, we always try to rehydrate these patients and to give pain management. Because if they're not rehydrated, you might think the clinical signs might be due to a bacterial infection, to inflammation, but in the true life, it's really true to the dehydration. And after rehydrating these patients and with symptomatic treatment, we assess the clinical signs. And if the patient, the dog, is still depressed or shows a tachycardia, tachypnea, or is hypotensive, then we think this is not due to hypovolemia, and we consider giving antibiotics because an underlying bacterial infection might be present. But usually dogs with acute diarrhea really respond very well on fluid therapy and pain management, and the level or these parameters might be in the normal range. Also very important, if we have to decide to give antibiotics or not, is to assess if you have a systemic inflammatory response on the CBC and biochemistry profile. So we look on clinical signs, of course. If we have an elevated rectal temperature, fever, then we think this is not due to an uncomplicated diarrhea event. We think this might really be an infection, so we would consider to give antibiotics in these cases. Also, if we see a very high number of neutrophils or a very low number of neutrophils, because on the one hand, we see this is an inflammatory response, and on the other hand, with a very low number, we know that we have an increased risk of sepsis if we have a barrier dysfunction and immunosuppression to a low number of neutrophils. In addition, if we see a left shift, a high number of banded neutrophils, we also know that this is due to an inflammation, and we would consider to give antibiotics, as well as in a hypoglycemic patient, because hypoglycemia can also be due to sepsis. And nowadays, we might also consider to measure the C-reactive protein, and if this is significantly elevated, this might also be a sign of severe inflammation and maybe bacterial infection. Another reason justifying to give antibiotics is always if we think that we have a barrier dysfunction in acute diarrhea. That means a destruction of the intestinal mucosa and the combination of immune suppression. For example, a dog with immune hemolytic anemia is on steroids, and maybe you start with mycophenolate, and this dog starts with acute diarrhea, maybe bloody diarrhea, and is immunosuppressed, then this might be a high risk of translocating bacteria, and the dog cannot react to this translocation, and this might be very dangerous, and the dog might become septic. And that's the reason why we would consider to start antibiotics in these cases. We also need a functioning liver, because we know the liver is the first line defense against translocating bacteria. And if we have a dog with acute diarrhea or chronic diarrhea, and for example, portosystemic shunt, we know that the bacteria cannot be cleared, and this makes a high-risk patient. And in addition, if you also see that we have a severe liver dysfunction, we can also assume that bacteria translocated from the intestinal tract might not be cleared, and again, we need to give antibiotics to this case. And there's another group of patients. We see sometimes that some dogs with acute diarrhea, very few dogs, might maybe not respond to symptomatic treatment and still show diarrhea after 7 to 10 days. And I think this is a very critical window, because this is the phase where an acute disease might progress to a chronic disease. And in these cases, sometimes nowadays we give a pulsed antibiotic treatment, because maybe there might be an involvement of bacteria and preventing that the dog is really responding to the symptomatic treatment. And we do not give long-term antibiotics to these cases, because we cannot make this environment sterile, so the GI tract will never be sterile. So the only aim is that we can suppress maybe some enteropathogens, and then hopefully the individual dog can handle the intestinal microbiota again, and we can stop the antibiotics. So we do not give long-term antibiotics in these cases if it's not needed, because we would create again a long-term dysbiosis maybe. So now we switch to a case with chronic diarrhea. This is an old German Shepherd dog, 5 years old, male neutered, and this dog showed chronic diarrhea. And it's always bad to hear for a clinician if this is for years. And this dog, most of the time, had a pasty consistency of his feces, but sometimes even watery diarrhea. The dog was on physical examination alert, everything was unremarkable. And so we performed a workup of these cases, and the CBC biochemistry profile was normal. We did an extended profile to rule out extraintestinal diseases like pancreatic insufficiency, Addison's disease, so everything was fine. But we also performed a fecal panel. That means we performed the fecal flotation, and the owner also showed us a fecal culture result of these dogs, because we do not perform fecal cultures in chronic diarrhea cases. So this is not from our submission of feces, but we had these results. And it was very interesting to see that that there was a culture of hemolytic E. coli, Clostridium perfringens, and Campylobacter. And also this laboratory provided sensitivity testing, for example, hemolytic E. coli. As you can see, this is in German, but this is the sensitivity testing. And this looked like a very resistant bacterial strain already, but fluoroquinolones would work. So now we have to decide, do we need to give antibiotics to this case with chronic diarrhea? On physical examination, the dog is unremarkably stable. Do we need to give antibiotics, maybe based on this fecal culture result? And actually, we performed a study where we wanted to know the diagnostic value of a fecal culture in dogs with chronic diarrhea. And this was published in the Journal of Veterinary Internal Medicine. And what we did was that we sent feces from dogs with chronic diarrhea and also from healthy dogs blinded to 3 different laboratories. And we, on the one hand, wanted to know if we can culture enteropathogens, so that we can know maybe or identify if enteropathogens in chronic diarrhea, if they have a role in this chronic process. And actually, we could not identify a lot of enteropathogens. The only bacterial strain which is considered to be a potential enteropathogen was hemolytic E. coli. But very interesting was when we looked at the results of the hemolytic E. coli, especially in lab 1 and lab 2, was that more dogs of the healthy control group, as you can see here in red, and more dogs of the healthy control group were positive for an hemolytic E. coli compared to the chronic diarrhea group. And this was the same in 2 labs. In 1 lab, there was nearly no culturing of this hemolytic E. coli. So there's a discrepancy between the labs, of course, but more important for us is that we can identify hemolytic E. coli also in a high number of healthy, normal dogs. So our assessment of this fecal panel in Merlin was that the hemolytic E. coli has very likely no clinical relevance, because we also see this bacteria in healthy dogs. And the same is true for Clostridium perfringens. This is a normal inhabitant of the large intestine. So if we can culture this, this does not reflect that we have an aggressive bacterial strain causing this chronic diarrhea. And moreover, also Campylobacter can also be diagnosed or found in healthy dogs. And we think as long as we do not see a systemic inflammatory response, we have not to treat with antibiotics in these cases. So our opinion at the moment is that in chronic enteropathies, as in Merlin, we do not have to treat with antibiotics in any case. We think chronic enteropathy means that we have a multifactorial disease involving the barrier dysfunction. That means if we have a disturbed intestinal barrier, the immune system will respond to the commensal bacteria and maybe normal dietary components. We know that sometimes we have to address the immune system with anti-inflammatory medications in these cases. We know that dysbiosis might be involved in chronic enteropathies. And of course, we know from human medicine, and also we see that some breeds are more likely to have chronic diarrhea. So we also think that genetic susceptibility might be one factor of a chronic enteropathy. And why do we not give antibiotics to these cases on a regular basis? You know, or maybe you still give antibiotics very frequently to dogs with chronic diarrhea. And we did this also some years ago. So we always performed a treatment trial with antibiotics in these cases. But nowadays, of course, we are afraid of creating a dysbiosis and producing resistant bacteria. And we recognize that we do not have to give antibiotics to all these cases. And the same is also in dermatology. For example, maybe 10 to 15 years ago, nearly every dog received with chronic dermatologic problems in bioderma, they received antibiotics systemically. And nowadays, they also changed. They try to treat the underlying disease and give some topical treatment, but we do not give systemic antibiotics to all these chronic cases in dermatology and GI diseases. And the reason is, in chronic enteropathies, we see a response in these cases. And in the literature, we have a response rate of about 10 to 20% if we give antibiotics. But usually we only treat the clinical signs because we do not cure these patients. And the positive response is most likely due to a reduction of the total bacterial load in these cases, and maybe more specifically, if we reduce the number of luminal attached bacteria. And if we reduce this number, then the immune system is not confronted with this attached bacteria anymore, and this might create a more stressful environment, and inflammation can go down. And that's the reason why maybe antibiotics might be beneficial in these cases, in clinical cases. But we see usually after stopping the antibiotics, if we have not treated the underlying disease, for example, food responsive disease, or maybe to give anti-inflammatory drugs to these cases, usually we see a relapse. For example, there's one study showing that in 33 dogs, they responded to antibiotics and all had relapsed after half a year to 1 year after stopping these antibiotics. So in my last few slides, I want to show our approach to these patients with chronic enteropathy. And the first step is always that we have to exclude extraintestinal causes like Addison's disease, like exocrine pancreatic insufficiency. We always perform imaging in these cases to rule out structural lesions, for example, intussusceptions, chronic foreign bodies, strictures. These are rare conditions, but still we want to rule this out. Then we always try to exclude parasites. So that means we do fecal flotations, plus we always treat these dogs because there might be occult parasitism in these cases. And then always the next step is to perform diet trials in these cases. Because in our institution, and we are really a second opinion and sometimes a third opinion clinic, we still see a response rate of 50 to 60% with food changes, with diet changes. So we have food responsive diseases very frequently. We always perform at least 2 to 3 treatment trials. We very frequently use commercial hydrolyzed diets. We see a high response to hydrolyzed diets, sometimes in combination with probiotics. And not so frequently we also perform, but this is more on a research basis, fecal microbiota transplants in these cases. But one of our first steps is really to combine a commercial hydrolyzed diet with probiotics, and we see a high success rate in these cases. The second step is, if there is no response, that we usually recommend to do endoscopy and histology of intestinal biopsies, because we want to know if there is a significant inflammation in the mucosa, and also the type of inflammation, if this really fits to an immune-mediated inflammation. And if we see a significant inflammation, we always treat with anti-inflammatory drugs. Most of the time we combine steroids with the second anti-inflammatory medication. But if we do not see a good response to the anti-inflammatory treatment, or especially if there is no significant sign of inflammation, we still use, in these very few cases, Tylosin, only to treat this potentially overgrowth or dysbiosis in these cases. And some dogs really respond well, and then we try to reduce the Tylosin and taper it down and stop it again. We still have indications for antibiotics in chronic enteropathies, especially if we have a bacterial infection of the intestinal mucosa. For example, this breed-specific disease like in boxer dogs or French bulldogs, this E. coli-associated disease, which is actually an immune-mediated disease, but the immune system cannot handle these E. coli strains. So we have to help and give antibiotics in these cases. And also if a dog with chronic diarrhea has signs of systemic inflammation or sepsis, very similar to a dog with acute diarrhea, we have to give antibiotics in these cases. At the end of my presentation, I want to show the outcome of our case Merlin with chronic diarrhea. And this dog really had chronic diarrhea for years, but this dog responded really to a hydrolyzed diet in combination with probiotics. And we also added some cobalamin to this case, because we saw that the cobalamin was at the lower end of the normal range, and maybe we have already some insufficiency on a cell level of cobalamin. So the key findings of my presentation is that acute diarrhea is usually a self-limiting disease, and we should really restrict the antibiotics to patients with a systemic inflammatory response on clinical signs, fever, for example, or CBC changes, or if dogs are not responding to the symptomatic treatment. We know that primary enteropathogens from our study do not play a significant role in chronic enteropathy, so we don't have to treat a specific bacterial strain with antibiotics in chronic enteropathy cases. But we know that dysbiosis is frequently present and might be responsible for this chronic inflammation. But still, we do not treat this dysbiosis with antibiotics. We always try to identify the underlying cause, to perform anti-inflammatory treatment, dietary trials, and in only very selected cases, we give antibiotics in chronic diarrhea cases, especially if we have not an adequate response to our treatment trials, if we suspect a true bacterial infection of the intestinal mucosa, or we have a case with signs of a systemic inflammation. I think this is a lot of information now, but I hope you enjoyed this lecture, and I'm very happy to answer any questions. Thank you very much.
And answer session of the Purina Institute Microbiome Forum, putting microbiome science into clinical practice. I'm sure you had 2.5 hours of wonderful learning and lectures, and now you have time to ask your questions from the eminent faculty that share with you their experience and knowledge. We received a lot of questions you submitted in the questions and answers session, and you can continue to do so. And without any delay, I'm asking Dr. Jan Suchodolski to moderate discussion and invite all the speakers to participate in that. Let's enjoy it.
I was muted. Thanks, Natalia. Yes, I would definitely echo that we had a really, thank you for everybody presenting. We have a lot of questions, so it shows there's a lot of interest in the microbiome, and especially clinical applications. So we really want to take our time and extend questions to all our speakers. And I wish that we had got all the nice discussions and really use some practical findings or new experiences. So let me start. There's a lot of antibiotic questions. So let me start with one. Can the microbiome be restored by nutritional support in developing puppies exposed to antibiotics? So maybe that's a question that I think clinically we don't know much yet, but maybe Gail, in some of your studies, do you have any experience? Can we restore the microbiome in developing puppies?
Oh, it's a great question. And like you said, there's a lot of room for more research in this area. I know we have data showing microbiome over time in puppies and showing how it's affected by weaning stress. And I know that we can affect that with probiotics and prebiotics. We haven't done studies with antibiotics per se. Mm-hmm. So maybe Linda, Mike has, oh, I'm sorry. I'm passing this on to somebody else.
Okay. Mike, do you have any experiences?
Yeah, I'd like to echo that's really a great question, that we do need a lot more data. You know, we believe that we can induce some clinical improvement, but whether or not the microbiome recovery correlates with that clinical improvement is still where we're lacking a lot of information.
Mm-hmm. I know, Stefan, and you have some experiences FMT on your clinical practice in Munich. This is also with young puppies too?
Actually, I think it's a very critical phase if you have a young individual and a puppy. So I think also it might be very similar to human medicine, that they might progress to chronic disease. So what we see frequently is in young dogs treated, for example, with metronidazole. And metronidazole might not be the big problem in adult dogs, but I think in young dogs, maybe you really can induce a significant dysbiosis and maybe chronic disease. And in these cases, we really sometimes consider to give fecal transplants.
Okay. So in line with that, do we know, do also injectable antibiotics have any or as much impact on the microbiome as parentally, or I guess orally administered medications? This question was open to anybody? I can give a little bit of background. So yes, we have a few studies. Actually, we have really studies. Most studies that we did where we looked really at the microbiome with oral antibiotics. But we have a few cases that we know also after injectables, there's also a huge dysbiosis can be induced. And the reason is that most antibiotics ultimately going to reach the small intestine to the liver, to the bile and so on, and that can also induce a dysbiosis. So yes. Dr. Gale, have you ever found that different strains of probiotics have an antagonistic effect on each other? In other words, is it best to supplement with few but effective strains rather than several strains?
That's a good question. I think it depends on what the strains are. I don't know that we've seen that if you take two strains together, depending on the strains, you can blunt the effect. I don't know if I've ever seen it totally disappear, but I think it just depends on the product. I mean, there's a number of good multi-strain products that are on the market that are efficacious. So it's unfortunately just one of those. It depends on what the strains are. Answers.
There was a lot of questions about dietary influence on the microbiome, especially dysbiosis index. So one question was, now also directed to me, have you ever looked at changes in the dysbiosis index with various diet compositions and types? And there's actually a follow-up question which follows that is, how do we determine what is and what is not dysbiosis, given the differences in microbiota in dogs, cats, fed kibbles versus canned diets versus raw foods? And I think that's a great question. And I think as we're learning with any other, this is my personal opinion now, and based on, I think a lot of data that we have is that I think there's obviously a huge range of normal, what is normal. However, we see very clearly that most diets, yes, when you look at just the microbiome itself or the dysbiosis index, don't make any changes in that. And again, because even if you know that there's plenty of studies out there that have shown that if you give fiber, for example, yes, a few groups change, but the magnitude of change is rather minor, especially when you compare this to antibiotic usage or GI disease, where we have a much, much bigger shift and larger shift in that. So again, I think we're learning definitely diet has influenced fibers on the micrometabolome, but it doesn't necessarily lead to a very big shift. And I think like also probably one problem with microbiome studies is that most papers have really compared one small thing to another small thing and never to the big reference population. And so now, since we have now markers that kind of define a reference population, we truly see that actually the influence is not as large as we thought it is. I think so. So generally, the dysbiosis index is not affected by those components. So there's much more increased antibiotic usage and in GI disease. A question for Linda. As a general practitioner presented with a dog or cat with diarrhea, what do you recommend as the primary workup and management of the case? And what would be the subsequent step if the previous one wasn't effective? So I guess, what are your steps in the workout?
It depends on the age of the animal and how, if I find any markers of severe disease, such as a low albumin, a low cobalamin, high disease activity. That's been shown in dogs, but the negative prognostic markers in cats are not as well defined. But look for severe disease, such as if the animal has lost a lot of weight, has a lot of diarrhea, or has completely stopped eating, or things like that. Then you need to be a bit quicker in your workup. But if you don't have markers of severe disease, which you know when you have done your basic workup, so blood works, and then I mean what is often called as a full profile or total profile. And if they've had problems for a long time, I always check cobalamin and folate. TLI if it's a dog losing weight and has diarrhea. TLI in cats if the cobalamin is low. Otherwise, it's usually not worth that. And PLI in cats if they are anorectic and have been vomiting on and off for a long time. And I always check for parasites the first thing I do, especially in young animals or dogs used for hunting or cats that are out hunting for prey. Then your likelihood of finding parasites is much higher. So the basic workup is checking for parasites, doing the blood works, and also ultrasound of the abdomen is one of the first things we do if the animals has had clinical signs for a long time. But in puppies, we often just start with a parasite test, including Giardia, because you often find the answer there. And if you don't have any evidence of severe disease or a marker for negative prognosis, then I start with a dietary trial. Often I go for hybridized diets. if they haven't tried that before, or if they are severely anorectic, we usually do a test trial. So we have open cans of kibbles, so we can actually serve kibbles to the dog or the cat, and sometimes that works for cats too, so they can choose what they find most interesting. And that's often a good idea, because then the owners are much more positive about buying that type of food. They often say that, oh, it's not going to work, my dog is not going to eat that. But if you actually serve different types of kibbles, then usually they show interest in one of them, and you can often send them home with that type of diet. And I often use a probiotic that is proven for chronic enteropathies in dogs. The same type of probiotic hasn't been proven for chronic enteropathies in cats, but it has been shown that in chronic constipation, it will improve the microbiota and also decrease inflammation. So you can say there's some evidence in the pilot study in cats too. So if I want to speed up the process, I start with a new type of diet and the probiotic, and then we go from there.
And if that doesn't work, what will be your next step?
It depends on how desperate the owner is and how the animal is doing. We could do another dietary trial. We sometimes try home-cooked diets, mostly for dogs. There are good basic hypoallergenic diets in clinical nutrition. So there are some recipes you could use there, or you can talk to a nutritionist about it, because sometimes the home-cooked diets work better than the hydrolyzed diets or the novel protein diets. If they don't want to go for another test trial, then we usually go for endoscopy and start some type of anti-inflammatory treatment with corticosteroids or cyclosporin or something similar. Unless the animal has diabetes, then you have to avoid the corticosteroids, etc. But I always try with a diet before I go for endoscopy, unless it is a dog with really low albumin that don't respond to a low-fat diet, then we want to rule out lymphoma. So we don't wait very long with those. But that's why I want to know if you have signs of severe disease or not.
Great. That brings me kind of to the next question that is quite often answered about owners want a quick fix with diarrhea and antibiotics and so on. So how do you see antibiotics in this workup? So maybe Linda first, and then Stefan, and then Jörg.
Yeah, we don't do the typical workup of antibiotic trials. So classically, you start with diet, and then if the dog don't respond to the diet, you go for antibiotics for 2 weeks. And if the dog responds to antibiotics, you call it antibiotic responsive diarrhea. And if that doesn't work, you go for corticosteroids or other types of anti-inflammatory treatments. And in Sweden, we have quite strict rules regarding antibiotics. So I haven't used antibiotic trials for, I would say, 10, 15 years. And there are so many dogs that do respond to antibiotics that also respond to other types of treatment. There are some dogs that truly don't respond to anything else than antibiotics, but there are very few if you skip that step. So the quick fix is to avoid the antibiotics if you have because you asked something about the quick fix too, didn't you? Yeah.
So yeah,
there are actually 2 studies that both were published in 2019 looking at metronidazole in acute diarrhea. So in the first, that was Langlois, published in JVIM in 2019. They had 31 dogs with acute diarrhea. They gave them a placebo or metronidazole. And the metronidazole, well, those dogs, the diarrhea stopped after 2.1 days, and with the placebo, 3.6 days. So the difference was extremely short, and that doesn't really justify to give a broad spectrum antibiotics that will kill a lot of the beneficial microbes just because you have 1.5 day less of diarrhea. And in the other study by Schmalberg, they had 60 dogs, and there was no difference in the length of diarrhea if the animal was given a probiotic, metronidazole, or placebo. So actually metronidazole is not that much of a quick fix that we often think as a clinician. So the quick fix for me would be to send the animal home with acute diarrhea with one of those clay preparations that usually contain some prebiotics as well as a low dose of probiotics, because they usually respond to that, most of them.
Mm-hmm. Stefan?
Okay, so maybe if we talk about acute diarrhea, I have the same opinion as Linda. And but I think it's more that we have this feeling of tradition to giving antibiotics, because maybe 15 years ago, when I was first in private practice, it was completely normal to give antibiotics to every dog. And so I think clinicians are very used to give antibiotics, but nobody was considering not to giving it or comparing it to placebo, for example. And now we have these studies showing that nearly every dog with uncomplicated, that means a stable dog without fever and all these inflammatory signs, an uncomplicated diarrhea case, they usually respond or recover in 4 days. And sometimes it lasts 7 days, but usually in 4 days they are happy again. So that means usually we do not need antibiotics. And I completely agree to Linda that we create maybe a dysbiosis by giving antibiotics, which we do not want. And so we do not give antibiotics on a routine basis. We always select these cases very thoroughly.
And in chronic diarrhea, what is your workup?
Yeah, in chronic diarrhea, I have also to admit that we treated several years ago nearly every dog first with also uncomplicated chronic diarrhea with diet, and then we had a tylosin trial or antibiotic trial. And in my feeling, we had good responses. And in the literature, we think about 15 to 20% of dogs will respond to antibiotics. And but I think that this is usually not a long-term success, because if you stop antibiotics, usually it comes back. And I think that we more or less treat the immune system, or we help the immune system by giving antibiotics, because we reduce the number of bacteria, we reduce the number of bacteria attached to the mucosa, and this is less stress for the immune system. So for the time, as long as we give antibiotics, everything is fine, but the underlying disease is not treated. And we stop the antibiotics and they will have clinical signs again. Sometimes we are lucky and we give antibiotics for a couple of weeks, and also the underlying disease might improve and they will be stable without antibiotics. But usually what we know from the literature and from our cases is that the diarrhea comes back when we stop the antibiotics.
So at which stage of your workup in chronic patients are you using antibiotics now?
Actually, I changed my opinion, because a few years ago, I always treated first with antibiotics and then with steroids. So we do the same workup as Linda, that we rule out parasites, do diet trials. We usually perform 3 or at least 2 to 3 diet trials, because I think that's very important. We modulate the intestinal microbiota by probiotics, sometimes by fecal transplants. And if they're not improving, most of the time we recommend to take biopsies. If we see a significant inflammation, that means a moderate to severe inflammation, and from the cell type, it looks like an immune-mediated inflammation, that means lymphocytes and plasma cells, then usually we do a treatment trial with steroids. But if it works, we reduce the steroids very quickly and add another immunosuppressive or anti-inflammatory drug, so that we do not see so many side effects. If we do not see a significant inflammation on the biopsies, or if they are not really responding well on the steroids, then sometimes in these selected cases, we try to give antibiotics and see the response. But these are really a few selected cases at the moment. And I think the same is in dermatology, for example. Many years ago, every dog with bioderma, they received antibiotics. And nowadays, they also treat underlying diseases. They use topical treatment, and they are not so focused on antibiotics anymore. And I think we can handle this, most of the cases, without antibiotics.
Jörg, what is your thoughts about the workup?
I show my age that I'm a little bit more positive antibiotics for chronic disease. Acute disease, we don't need to talk about it. I mean, Stefan in his studies and others have shown very well that there is absolutely no benefit of antibiotics, and if anything, that's detrimental. For chronic disease, it's a lot more complicated because I think, on one side, of course, Jan, you've shown that tylosin leads to dysbiosis, but I think Westermark studies are still very powerful, who showed that there was groups of dogs that didn't respond to anything else, that responded to tylosin, and when you took them back off the tylosin, they had a remission. They went out of remission, and so I believe that for my cases, the sort of chronic cases that don't respond to diet, which 50 to 60% of them do respond to diet, so that takes care of a big number of them, and then you take out the parasite ones at the very beginning, even before then, it doesn't leave that many cases. And I think, and I wouldn't call it an antibiotic trial because I don't use anything but tylosin. So, and we're not really sure, how much of tylosin's action in improving these dogs actually is truly an antibiotic effect. It could be an immune modulating effect. We don't really know what the effect is. But I do believe tylosin has few side effects. Yes, it does cause further dysbiosis from a scientific point of view, but I do see a lot of dogs that respond to it. And if you treat long enough, 6 to 8 weeks, many of them stay in remission. So I know I'm sort of, I'm probably a little bit, stubborn on that. Certainly FMT is a good alternative to that. And I'm recommending that more and more. But again, a lot of that is not known. What in fact happens and why tylosin improves these patients, I have no idea.
Mike, you raised your hand.
Yeah, just a comment back to the metronidazole addiction that many people of my age group had, earlier in our careers. Part of that stemmed, of course, because of the high prevalence in most areas of giardiasis as one of those differentials. And so metronidazole was a go-to. And we've actually, I think I mentioned in my talk, a Fenimore paper where it's ingrained, like the shelter that we had studied recently for nonspecific small bowel diarrhea, they wouldn't stop giving metronidazole. And so if we wanted to do a study in that shelter, we needed to find a way to gather data without changing their current protocol. And that's one of the Enterococcus faecium strain SF68 studies, where we just did the nonspecific diarrhea dogs with diet change and metronidazole, and then half the group got the probiotic as well. And, I always call it getting off the toilet a little bit faster. We did show an effect of a clinical effect of that treatment. And then just to remind the listeners, Jan's team and then Jorg's team, we coupled together to show if you were really going to do a nonspecific diarrhea challenge with antiparasiticide, that actually using benzimidazole makes more sense in these days because Bantel and fenbendazole used for giardia and then the nematodes don't change the microbiome with the tools that we use in that study, whereas metronidazole obviously does. So back to Linda's chat about, doing the parasite trial. As a parasitologist, I love to do diet plus fenbendazole in the cat or dog, or diet plus Fabantel in the dog as my therapeutic trial with or without probiotic before going on, to the antibiotic trials.
Mm-hmm. Linda, you have one more comment to that?
Yeah, I'm just trying to unmute myself. And of course, I do use antibiotics at some time points, but I think we should save them for the last step. So I think really you should try all the other options first, and also thinking about the One Health perspective. We don't want to create antimicrobial resistance in the pet that can actually spread to the owner. So really, I think we should save the antibiotics as the last step. And then the ones that are truly antibiotic responsive, they won't respond to corticosteroids, but actually quite a few dogs will respond to metronidazole. When you stop the metronidazole, they will get diarrhea again, and some of them will respond to corticosteroids or other immune modulatory treatment or FMT. So my point of view here is trying to save the antibiotics as the last step for the chronic ones.
Stefan, last word to this question, and then we...
Yeah, I just wanted to say that I'm happy that Jörg mentioned that tylosin is working, and I think we need the whole information from different clinicians. There's not one truth answer to it at the moment. But I wanted to stress a little bit that sometimes in Europe, I hear that metronidazole is a very small spectrum antibiotic. And this is true, because if you want to treat a septic patient, it will very likely not work because it works against anaerobes. But from the view of the GI tract, we should know that most of the bacteria are anaerobes. So we have a very, very strong impact on the intestinal microbiota, and we kill a lot of anaerobes. So, and this might be also one reason why this antibiotic is working so well, because we reduce the bacteria. But it does not mean that this is a small spectrum antibiotics and has not a very impact on the body.
Mm-hmm. This is perfect because it answers two more questions. That's, can metronidazole or Tylenol administration lead to a decrease in the good bacteria like Yersinia nonnis? I think yes, it does. We know that. That has been nicely shown. So it definitely decreases the number of good bacteria. And the question was, why does metronidazole seems to work so quickly for many dogs with diarrhea? That's a question for Stefan. Is it due to some anti-inflammatory action or an antibiotic effect? I think we answered the question to some degree already. Your thought was that it's decreased the total number of bacteria, and because of that, maybe the immune system responds, but generally it does not really improve the microbial pattern, correct?
I think it would be too simplistic to think that we kill the bad bacteria, the harmful, and we save the beneficial bacteria. I think that's not working. And yeah, so I think that this is less stress response of the immune system to the lower number of bacteria attached to the mucosa. And I think that's the main mechanism.
Great. Let me ask a question to Holly. I think it's a great question about low diversity in bats. I think very actual question. Is it related to the high prevalence of viruses in the GI tract? Is there a link?
Yeah, that is an excellent question, and it's a really exciting area of research. Some of the hypotheses that we've been putting out are that bats may not be as dependent on services provided by the intestinal microbiome, and this may have influenced the evolution and adaptation of their immune systems. So perhaps they're not forming such tight links and relationships at that epithelial barrier in the gut. And if that's the case, they may be exhibiting more generalized responses to symbionts that come through their system. This could actually impact the relationship they have with viruses. So if they're having a more general response and sort of keeping the microbes at bay, they may be having similar responses to viruses and sort of allowing them to be transient members of the community that pass through. There are several other hypotheses about why viruses in bats are so pathogenic when they do transfer over to humans. So I just want to make a couple of notes here. Bats don't actually harbor more viruses than other groups of animals. They're just very diverse. Bats are very diverse, and so they have a proportional diversity of viruses as well. That said, the viruses that do move from bats into other animals tend to be more pathogenic. And one of the reasons we think that might be is due to the evolution of flight, which produces a lot of free radicals, and this has led to some unique adaptations in bats that allow them to repair DNA. They have like unique DNA repair mechanisms that are different from other animals. And this kind of creates an arms race in some ways between viruses and the host. And so viruses that are evolving to infect bats are actually able to persist in much stronger ways than they would in other organisms. So when they jump into a different host, they find it a lot easier to infect and proliferate. Coming back to the role of the microbiome, yeah, I think I kind of said this at the beginning that we don't really fully understand how this is working, but I think it has a lot to do with their immune systems. I'm not sure if that really answers the question.
No. It's a great answer. But to follow up on that, do we see any significant differences in fecal microbiomes for same species, but present in different regions and continents? So I guess.
Yeah, so this is seen in humans. It's seen in our domestic pets. You can very clearly see signatures of geography and distinguish populations based on where they're located. That said, the functional profiles of these microbes of the microbiomes are very similar. So you can think of it as, whether the species vary from continent to continent or, coast to coast, the species or the strains might be different, which is what allows us to distinguish where you're from. But the function of the microbes is likely to be very similar within species.
Mm-hmm. Yeah, so I just want to add something. There's a question about dysbiosis index in different countries and continents. Yeah, we looked at many different countries and continents, and we actually don't see a difference in the microbiome. And again, because the index itself, again, we need to understand we're comparing it to a diseased population. So again, there's a much larger shift in disease than you would notice in some country dependent. So we have different countries and we don't really see a significant difference. And again, maybe also in dogs and cats, I think many of them are on commercial dog food diets, so they're probably more similar in composition, whereas in humans, obviously, there's a huge impact on dietary differences between Asian countries or European countries or Western countries and so on. I think that's the reason. Diets. Let's talk about probiotics again. Gail, is there a good resource listing specific beneficial bacteria and the corresponding effects of those bacteria?
Oh,
hence,
that would be, yeah.
Hence, would there be sorry, just to finish.
Mm-hmm.
Hence, would we know if a probiotic is more anti-inflammatory or immune stimulating?
Yeah, I think the best place to get that information would be from the manufacturer. I haven't seen a overview presentation for probiotics that are out on the market for pets. There's a little bit more information for the human products, but I've never seen one that goes through their immune stimulating or anti-inflammatory properties. So unless somebody else on the panel has seen that type of thing, it'd be nice if it existed.
Yeah, I'm not aware of. Maybe a follow-up question. How do you measure transient versus permanent colonization
That's
in probiotics?
Yeah, basically what you do is you feed the probiotic. You have to have a test to be able to recover the probiotic from the feces, and you have a test that's specific to that strain. And basically what you do is feed the probiotic, get the fecal samples for various amounts of time after you stop feeding it and see when it disappears from the feces. Mm-hmm. So you can tell whether or not it persists for a long, long time, or if it's gone after 3 or 4 days after stopping.
Mm-hmm. To follow up on that, is it important, is the adhesion property important for probiotics? So that means, is it essential for them to be effective?
That's an excellent question. It's something that's difficult to measure non-invasively in vitro, or in vivo rather. So I would say we probably don't know for the probiotics that we have, whether there is adhesion or not in the live animal. I think it probably depends on what the end benefit and the mechanism of action of that particular probiotic is, whether it's acting by, secreting bioactive substances that are having the benefit, in which case then adhesion might not be so important. If it's competing with pathogens, it probably depends on how it's doing that. For example, SF68 produces bacteriocins, and it might not actually have to have adhesion if you have a probiotic that's competing for binding sites in the GI tract with pathogens, then it would probably be more important.
A follow-up question for Holly. Also, individuality in species, would this also affect probiotics? So do probiotics have to be species specific, do you think? Or can we use one probiotic that works in one animal or human and use it in other species?
Yeah, that's a great question. And I know that there are a lot of products that are marketing things like bifidobacterium for canines. And I think something that's important to keep in mind is that these groups, when we speak about them at the genus level or even higher levels of classification, they can be incredibly diverse. Even if it's a specific species, you might have different strains. And so I believe, and correct me if I'm wrong, that it won't be harmful to your animal, to the dog or the cat, to give them, a probiotic that maybe is something similar to what we're taking, but you're going to have limited benefit if it isn't species specific, and taking into account also just the the individual variation of different breeds or the lineages of these animals, I think that you can fine tune it. I think it's important to fine tune it. And I don't think we're there yet because we don't have enough information or data on how a lot of these probiotics are actually benefiting the host.
Mm-hmm. Gail, your thoughts?
Yeah, I think it's something like Holly was saying, there's a lot of information we don't know yet. I know most probiotics I've looked at have efficacy across a variety of species. So for example, SF68 was isolated from a human baby, but most of the efficacy studies are in livestock and in pets and in adult humans, so they can have cross species benefits. Probably depends on what the benefit is that we're looking for as well.
Great. So that's for Professor Lappin. Is every Enterococcus faecium SF68 the same? I found this strain also in products for large animals. I guess that's what Gail mentioned. However, there was a different NCIMB number. Can I trust in a probiotic called E. faecium only?
Yeah, now again, it'll be a great one for Gail to add on here,
Okay.
just because of the registration terminology, et cetera. But certainly, as Holly's already mentioned, genus and species, there are strain differences. One of the questions that's interesting about SF68 is, as it stayed the same, over the 40 years, since it was initially isolated, that if it's strain SF68, then in theory, it would be the same organism, but you couldn't trust another product that just says Enterococcus faecium. It may or may not be even remotely related to that organism, strain, or its effects.
Mm-hmm. Gail, you want to add to that?
Yeah, I would just say, basically the same as what Mike said. SF68 is a registered trademark. So if it specifically says SF68, then it would be produced the same way that the SF68 is produced for pet products that we use in Fortiflora. But I know that from what we've heard from the manufacturer of that probiotic, that you can affect its efficacy by how you ferment it. So if you had a different Enterococcus faecium, it wouldn't necessarily work the same. And there are pathogenic, as you all know, pathogenic strains of Enterococcus faecium too. So personally, if I was feeding an Enterococcus faecium probiotic, I'd want to make sure that it was one that really is a probiotic and not just one somebody isolated out of feces and then grew up. And then it's like.
Yeah, great.
Yeah.
So, Mike.
I just wanted to go back to the previous question about the will the same probiotic give the same effects in an alternate species? I think since we don't really know, it's really puts it upon the companies that market them for sale anyway, that they show efficacy, in the target animal species for those of us that buy them or teach vet students that these are the ones that work. It is actually quite important, I think, to have target animal efficacy data. And just one of the ones I think I mentioned briefly at the end of my talk was looking at Bifidobacterium longum, 999, coming from a dog launched product and then applying it to cats and seeing some of the same effects. So I guess I would be hopeful that many of those organisms would behave the same, and at least dogs versus cats. Now, whether or not dogs versus dolphins or dogs versus Gale, could certainly be different.
Yeah, that's a great point. So we are already there's two major areas that I still want to ask a few questions. So one of them is diagnostics and then fiber. Those are major questions. And a question for Dr. Steiner, for Jörg. What are the approximate specificity, negative predictive value, and positive predictive value of cobalamin folate?
If we only knew the answer to that question, there is no, I mean, the best studies that were done on this were done in would say the mid 80s. Devonport did some of them, and there's some other people who looked at that. And the best I can tell you is that the sensitivities and specificities in those studies were all around 50, 60%. Nobody, as far as I'm aware of, has ever looked at positive or negative predictive value. And clearly, it's not stellar. It can't be stellar with sensitivities and specificity of 50 and 60%. it is until the dysbiosis index, it was the most practical and best we had. But clearly that is no longer the case. But yeah, unfortunately, we don't know that. Especially the low cobalamin has many other conditions, obviously, that would also lead to a low cobalamin, any kind of severe and long-standing small intestinal disease that involves the distal small intestine. A high folate's a little bit different, probably a little bit more specific, simply based on the fact that other than supplementation, so some owners may supplement and you may not be aware of that, will obviously lead to an increased folate concentration, but really nothing else biologically will lead to an increase in folate. Now, but then the question is, how sensitive is it really? And what level do you use? If you just use the upper limit of normal, let's say is 15 and you use 16, that's certainly not going to be very specific. But if, we see dogs and cats that have serum folate concentrations of 30 and 40 and 60, and well, that becomes a little bit more suggestive. But yes, those are not very good diagnostic tests. Their sensitivity and specificity are both lacking. And I wish we would have more data on them, but we don't.
Mm-hmm. To follow up on that, also a question for Dr. Steiner, but then also for some other panel members. Oh, sorry. When is it appropriate to run a fecal PCR panel for a dog with chronic enteropathy, and how should a positive result for Clostridium difficile or Clostridium perfringens be interpreted? Jörg?
Well, I think those are really, that's a hard question to answer, because I think what we see at the referral practice, we see cases that have already been dealt with for quite a while. And so we probably run more of those panels than you would need to do in a primary care setting. But personally, even then, I don't run the panels very often. I run them when I have diarrhea that doesn't respond to the traditional workup that Linda has very nicely laid out, and I go by exactly the same thing that she laid out, with the minor difference in antibiotics as we discussed. But other than that, it's exactly the same thing. And so if I have bloody diarrhea where I can't find an underlying cause that's chronic, I certainly will run one of those. The other question is a even more difficult one. What do I do with a positive difficile or Clostridium perfringens? The difficile one, I really, very little evidence that would suggest that it's truly a primary pathogen in dogs and cats. So I try to look for another cause in those patients. So I usually do not recommend treating them. And perfringens, it really depends on the species, on the strain. So just any measuring a toxin is very important, and the only one that's really has shown any sort of what Stefan has shown is the NetF that has some agreement with what disease is actually clinically significant and with bloody diarrhea, but all the others are probably not really significant. And most normal animals have Clostridium perfringens. Most normal dogs, normal cats.
I might just add to that, because also was addressed to me the question. So we have a very strong correlation between the presence of Clostridium difficile and abnormal Hieranones, so low Hieranones, abnormal bile acid conversion, which also makes perfect sense, because it has been very nicely shown from human medicine, but also in vitro, that they are really very strongly interconnected. So alpha Clostridium difficile is truly, also my strong opinion, in most cases an overgrowth bacterium, because you have a dysbiosis to the underlying GI disease, you have no Hieranones, no bile acids, and as we see it very quite often. There's a few case reports in veterinary medicine, mostly younger dogs, where they confirmed the toxin per ELISA, but those are very rare cases. So just PCR would indicate more dysbiosis. In selected cases, you may, if you rule out everything else, maybe consider running a toxin ELISA to confirm that it might be the rule, but it's very rare. Since we're running out of time, psyllium and fiber, there was a lot of question. Linda, how do you determine a starting psyllium dose? And there was just that the 5 milliliter twice daily was a confusion, I think, to the listener.
Well, as I said in my lecture, there are very few studies on fiber in diseased animals. There are quite a few studies in healthy animals, but very few in diseased animals. Still, we know that fibers can be very beneficial. So the dose I found somewhere, I think that was in the very old Strombeck's manual of small, well, whatever it was called. It's not available any longer. It was sort of the Bible for gastroenterologists in the 90s, but it's not available any longer. But there they said 1 to 6 tablespoons of psyllium per day. So that's a huge difference. So I usually start quite low, and when I start, I tell the owners that most of these dogs will respond, but some will actually get worse. So that I usually start lower than that. So that border terrier I showed in my talk, she got 10 mils, which is 2/3 of a tablespoon per day to start with, just to see that they don't get much worse. And then you can taper up the dose. And usually if they get more than they can actually tolerate, the feces will be like a corkscrew appearance, which will fascinate the owner a lot. But if you decrease the dose, then they will get back to normal feces again. So you start somewhere and then you can taper it up.
Mm-hmm. So a follow-up question for Jörg. Would you try psyllium before tylosin?
Well, psyllium is sort of a very special fiber, and I think it's important to hold those two different, all these different fibers apart. The psyllium is a non-fermentable fiber, so that mostly has an effect with colonic disease, versus, if you have a fermentable fiber like fructooligosaccharides or some mixture of, whatever, inulin or whatever, that has a much bigger impact on the microbiota, as it's a substrate for microbiota nutritionally. So I don't use psyllium very often. Mostly with colonic disease, isolated colonic disease, much more likely to use a prebiotic fiber, so a fermentable fiber.
Mike, I know you have some studies that you presented at ACIM with psyllium. Can you give us a background?
Yes, the most recent psyllium studies from our group have actually been with the Fortiflora SA product and the amount of psyllium that's added to that probiotic, and we'd shown potentially lessening severity of clavamox diarrhea, as well as using it for a treatment of clavamox diarrhea. And then most recently with mycophenolate, when we get into treating those IBD and other immune diseases with immune suppressive agents, mycophenolate runs in that 20% diarrhea rate, and so I have not done psyllium alone, but probiotic plus psyllium, we were able to lessen severity of diarrhea in those mycophenolate dogs. So it becomes something to consider as we're treating our IBD dogs, is that could we perhaps have a positive effect on the microbiome and lessen toxicities of some of the drugs that we're using to treat that condition.
Since we heard today that a lot of things change over time, Stefan, what is the current thinking about fasting dogs with uncomplicated diarrhea for 12 to 24 hours?
These were all great questions, and this is also a good one. And yeah, actually, I think that this is also that we feed them earlier, and I know there are some studies also from York showing in Barberosis cases that the barrier function is improved by early feeding. And I think nutrition for the enterocytes is also very important. So not giving diets to a dog with acute diarrhea is maybe only cosmetic, so that nothing is in the GI tract. But I would not give too much food at this time, of course, because we know this from ourselves, that if we eat too much, that's maybe causing more problems. But I need small amounts frequently, and of a highly digestible diet is very important for the enterocytes, and maybe some fibers for the colonocytes as energy source. I think that's important.
Mm-hmm. Quick question to follow up, again, infectious agents. You mentioned, Stefan, you do not treat Campylobacter species. What about Campylobacter jejuni?
Actually, that's my philosophy. I do not treat a fecal panel. I always treat the patient concerning antibiotics, And for example, if I have a patient with a potential enteropathogen, I would maybe in this chronic disease and stable, I would rather consider to change the diet, maybe probiotics or fecal transplants to displace this pathogen or potential pathogen. But concerning antibiotics, I always treat the patient and I look for the clinical signs. So if I have an inflammatory response, if a dog is not responding to the diet and all other treatment options, then and it has an inflammatory response also on blood work, then in these cases I would treat with antibiotics. But I do not treat if I see, for example, Clostridium perfringens or Campylobacter or Salmonella, because in dogs there might be a bacterial strain without creating any problems to the dog.
As a follow-up question to Holly, how is Clostridium perfringens classified? It belongs to the Clostridium genera, but is not considered a beneficial bacterium.
Yeah, I think that this speaks to my earlier comment about the classification and taxonomy of these microbes. You know, there's a lot of diversity below even that species level and the strain level. So Clostridium is a huge group, and many of the species that are in that genus have very different modes of action and different metabolic pathways that they utilize. And so you can't just say because it's, you know, a bacterium in this group that it's necessarily good or bad. I think we were thinking about these as whole communities too. And yeah, some may be pathogenic. It may also be context dependent. So, I mean, perfringens is actually frequently found as a part of the regular microbiome. It has more to do with the relative abundance of this microbe, probably in many cases. Then you may find a positive PCR test and you may see that it's there, but it doesn't necessarily mean that it's causing a disease.
There's a question follow up on that, Holly, and maybe Mike knows more about also something. The relationship between intestinal worms and dysbiosis. What do we know about that?
That's an interesting area. I don't know much about it, but I think that there is a lot of research showing that the immune response to helminths or other intestinal parasites may have an indirect impact on the immune response to microbes as well. And I think there's even some evidence to show that helminths can directly influence the microbiota through their own metabolic pathways. Maybe someone else knows more than I do about this.
Yeah, I can't add much to that, but I would like to go back to the Campylobacter issue. We have had multiple drug-resistant Campylobacter going around the United States for over 5 years now that sickens owners as well as animals. And so it is quite important to remember that oral antibiotics and perhaps even parenteral can select for resistant strains of bugs that we just kind of routinely thought we could give them Altylosin or other drugs to, and then affect people in a One Health fashion.
Yeah. On that note, with the One Health, I think people here are probably aware that research has shown that our domestic pets and the owners often actually share microbes in the home environment. So we have to really think about how what we do with our pets or what we do with ourselves is moving in both directions.
Linda, you had some comments?
Yeah, first on Campylobacter, that if you infect 8-week-old puppies with Campylobacter, half of them will develop self-limiting disease. And it's very, very common especially for dogs under the age of 2 to have Campylobacter jejuni without having any clinical signs at all. So just because we find it doesn't mean that it's actually a problem. And secondly, there was a really nice paper that came out just recently on Giardia, where they had dogs that had very little symptoms or clinical signs of Giardia, but when they followed the microbiome over several months, they could show that they had a dysbiosis and it actually got worse over time, and they followed them for a few months. And so at least for Giardia, there seems to be some evidence that it can create dysbiosis.
Yeah, I saw this paper and I just want to highlight one thing that we, again, need to be a little bit careful, because many microbiome papers really just compare small effects. So that's important to always highlight. So when we see something cause a dysbiosis, we need to be really careful in overinterpretation. Doesn't mean that there's no changes, but again, many of those papers don't put this in a big perspective of what is the range of normal. So it can be a small shift. So I'm gonna just kind of highlight that. A few questions.
Jan, I think our time is coming to the
Okay.
end because it's, you know, the discussion about microbiome can go on and go on because it's a such exciting topic, and I really apologize to interrupt that. And but it's getting even more exciting because we have participants who are attending this conference from the different parts of the world, and we have such a great international faculty that is answering those questions and sharing experience. So I really hope that some of those questions still can be addressed tomorrow, because we have tomorrow another day, and not even maybe more exciting program for tomorrow, and those questions could be there. Also, you could visit the purinainstitute.com, where we designed specifically for veterinary and healthcare professionals the Purina Institute Microbiome Forum, a platform where you can find the foundational knowledge on the microbiome, where you can find the proven nutritional interventions, and that may help you in your clinical practice. And with this, Jan, if you have something to say, and then I come with certain announcements which important for the day tomorrow.
No, I think thank everybody for really a lively discussion. We have a broad range of topics. We have a broad range of expertise, and I think it's really great. I think, as you can see, we know already something how to bring the microbiome into context. I guess we all probably agree that prevention is the way to go. The microbiome is an important organ for health benefits. So I think at this stage can we prevent it? That's probably the easier way to go. We still have to learn much about how to bring it back to normal. But I think I really love all those questions. Thank you, everybody, for bringing those questions. Sorry that we couldn't answer all of them. We're going to try to answer a few by email or by text. Some of those questions, especially about fecal transplants or microbiome dysbiosis, is going to be hopefully answered tomorrow, because we have a lecture about fecal transplantation tomorrow. And I'm hoping that you can join us back tomorrow.
Yeah. Thank you very much again for everyone. And I know that it was a little bit too much time to ask from you. For those who was not able to see all presentations today or want to revisit it again, you can find them on the same platform, because all lectures will be available on demand. Also on the place which calls resources, you can find the list of the references that have been used during the presentations today. And another important point, that you can find the downloadable decks that has been presented today. If you go to the agenda and you see the names of their speakers and the titles of their presentations next to that, you will see the downloadable slides, which I believe would be very helpful. And with this, I really appreciate all your time, all your effort, all your knowledge, and the passion to talk about the microbiome, and looking forward to see you tomorrow. Thank you very much.